I want to take this opportunity this afternoon to welcome everyone to the 13th Biennial ASF Conference, Championing Progress. It's really great to see everyone here, to see all the individuals with Angelman syndrome, the families, the care providers, the researchers, clinicians, educators, and everyone that is here to support all of you. It's a tremendous turnout. We're so happy to be here. We hope that you've already had a chance to enjoy Orlando, and we really hope that you have a fantastic week. I wanted to introduce myself. My name is Eileen Brown. I'm the Executive Director of the Angelman Syndrome Foundation. And I wanted to tell you just a little bit about myself and my family. My husband, Tom, and I, and our family live in Aurora, Illinois. It's a suburb of Chicago. We're about 45 miles straight west of downtown Chicago. And we have 4 children, and from left to right, our middle son, Jonathan, he is 16. Joe is 19, and he'll be a sophomore in college. My husband, Tom, and to the very far right, Kimberly, our youngest daughter. And then our oldest daughter, Caitlin, is 22, and she has Angelman syndrome. Just a little bit about Caitlin. Caitlin is, in many ways, very typical of individuals with Angelman syndrome. She loves water. She can be very happy and engaging. She just exited the school system, the educational system, and as such, she is now working. She has a couple of jobs. The jobs that she particularly likes to do, as you see starting on the bottom left, she goes to a business in our area. She does all the labeling of their bags for them. She also does some shredding, and she also cuts all the raffia ribbon for the bags that they use in that business. She's also started to do some inventory for them, so unpacking their inventory and putting the products on hangers and helping in sorting in that way. She's incredibly proud of her work and what she does, and we are incredibly proud of her as well. So for those of you that might be wondering, Caitlin is deletion positive. She has been ambulatory and walking independently since she was just about 3 years old. She did have some seizures when she was young and was off all of her seizure medication for about 10 years. And just following the onset of puberty, she did have one episode of status epilepticus, and she has been very, very well controlled on medication, but one of the things that has helped her considerably is the low glycemic diet. And for those of you that are interested in that, there will be a speaker that's going to be talking about that here as well. So overall, we've enjoyed a very wonderful and blessed life, not only with Caitlin, but with our entire family. And I just wanted to share a little bit with you about my family as well. Just as I get a chance to know you and all of your families, I hope that you get a little chance to get to know me and my family as well. One of the things that I want all of you to know is that the Angelman Syndrome Foundation is here for you. This is your organization, but we're here to help and support you. And being here in Orlando is very, very special for me because it was 20 years ago at the 1993 conference that it was my first opportunity to be sitting where all of you are. And that first experience for me and my husband, we were very excited to be here, but we were also very scared because this was a whole new world for us. It was something that neither of us had ever dealt with on either side of our family, and it was, quite frankly, a world where we did not want to be. But what we felt and what we experienced at the conference was an incredible sense of relief, but also a sense of belonging. We felt like this is where we should be, and we were with the people who knew and understand and understood everything that we were going through. And that has carried through for our family with this community and with the foundation. And we hope that and it's our goal and our mission as an organization to provide that same kind of care and support for all of our families within the Angelman community. So while you are here, I want to encourage you all to attend as many sessions as you can. You'll learn a lot. And I also want you to spend some time networking with each other because you're going to learn even more. So, on behalf of the staff and the board of directors of the foundation, I do want to take the opportunity to welcome you all. So thank you all for being here. We hope that you have a terrific, terrific conference experience. At this point, it's really my pleasure to introduce to you our newly elected ASF president, Mr. Greg Dorman. Greg and his wife Natalie have 3 children, and their oldest daughter, Danica has Angelman syndrome. Greg and his family reside in Stockton, California, and Greg has served on our board as a director, as vice president, and he now serves as the president of the board of directors. Greg?
Welcome, everyone. I want to thank you guys for being here, and I also definitely want to welcome our newly diagnosed families that are here for the first time. We've all been in your shoes. We understand what you're going through, and we are here for you, as Eileen pointed out. So today, it's my job today. I get to thank quite a few people, but as many of you have recently read, the foundation was recently able to put aside about $1.3 million towards research. And we're able to do this because of the success of our annual Walkathon that's held in May in 30 cities across the U.S. And the support that we receive during those walks is because of all of you, and we thank everyone in this room for all of the support. It's a lot of time and effort to put those walk sites on. Having done it for 6 years in Sacramento, I understand the commitment and the time that it takes. And so my job today is to thank 6 people that were the organizers of the walks in their particular cities. I also do want to thank folks that are on the committees of these walks and also the folks that attend these walks. They're great events, they're great networking events like this is here, but they also are a fundraising event. And so my job today is to thank 6 of those people. So if they could please come up, I'd like to give each person a little thank you. The first is Leanne Boyce. Next is Davar Burbidge. Next is Susan Ravelette. Grace Sampson. And Cindy Snyder. And last but not least is Lizzie Sordia. And also what I'd like to do, I'm not sure, is Mitch Caspert here by chance? Okay, I don't think Mitch has made it in yet. Mitch is a long-term member of the board of directors. Mitch's term is up, and so I wanted to thank him. Unfortunately, he's not here yet. He's flying in, I think, later today. So I did want to thank Mitch publicly, and of course we'll thank him in person as well. Secondly, and most importantly, 4 years ago, the organization voted Tim McCarty as our president of the board of directors, and it was in Orlando 4 years ago that he agreed to do this. And as many of you know or may not know, the board of directors here for the organization is an all-volunteer board. And all of us, many of us, maybe with the exception of 1 or 2, are parents. So the time commitment that we put it forth is, it's on our own time. We all live the lives that you live. And so it is. It's a very challenging job. And so Tim agreed to do this 4 years ago. And if you look at where the organization has gone in these 4 years, we've been able to open 2 clinics, one in the University of North Carolina, one in Boston. You look at where the research has gone, and although Tim can't take all the credit for the research, we'll give a few of these folks some credit for that. Again, it's under his leadership. And it's because of you, under his leadership and your support has put us on the path where we are today. So I just want to say, Tim, thank you very much. If you got to stand up, buddy. So again, thank you very much for being here. I'm going to turn it back over to Eileen for a few more awards.
I do want to take the opportunity to introduce those board members that are here. It's really important for all of you to know, and I mentioned this earlier, that this is your organization, and we want you to be able to provide your feedback, not only to us on the staff, but also to the board members as well. And I know that sometimes it may seem like you can't approach the board members, but that's absolutely not the case. And you may have seen their pictures and their names on our website, but I would like to have them stand and be recognized and introduce them all to you. So Susan Ravelette from San Diego, California. Kathy Rokita from Indianapolis. Greg Dorman from Stockton, California. Frank McCullough from, oh my gosh, it's just slipped my tongue. Charleston, West Virginia, sorry. Dan Harvey from San Diego. Dr. Charlie Williams from Gainesville. And John Sugden, are you here? John Sugden from New York. Tim McCarty from Hinsdale, Illinois. And I'm not sure, did I see Steve Katz here yet? Okay. I know that Steve will be here as well. So please, by all means, questions, comments, concerns, please stop one of the board members or one of the staff members. We're here to help you and answer any questions that we can. So, thank you. The Angelman Syndrome Foundation presents 2 awards at our biennial conference. And we are very, very privileged today to present the Harry and Audrey Angelman Award for Meritorious Service. And this particular award is for those people that have really demonstrated a very strong commitment, not only to the organization itself, but also to the disorder, and have also really promoted Angelman syndrome awareness. And this year we are presenting this award to co-recipients of the award. And the first person that we are going to be presenting to, I think may be tied up or not here yet. So we are going to go ahead and present to the second recipient of the Harry and Audrey Angelman Award. I've been incredibly privileged and honored to know and work with this individual for a number of years. This person does not have a child with Angelman syndrome, but he does in fact have a sister who has Angelman syndrome. And his story is a story of a lifetime of commitment to his sister. His sister was not diagnosed with Angelman syndrome until she was 55 years of age. After his own parents' passing, he assumed the responsibility of care for managing her care and all of her affairs. His involvement with Angelman syndrome truly simply could have stopped right there, but it didn't, and it still hasn't. He's taken on Angelman syndrome head-on, and he has been very involved and influential with the Angelman Syndrome Foundation Board of Directors. and he has served in many capacities. He served for 8 years on the board of directors. He served 4 of those years as the ASF treasurer, and he served 2 of those years as the ASF vice president. He's worked on a number of task force and committees while on the board, and he has and is currently serving on the finance committee of the organization. And he's done this for nearly 10 years. In addition to his work on the board, he has also successfully served the Angelman Syndrome Foundation and our AS community since 2005 as the Washington, D.C. Walk Coordinator. And under his direction, he has helped to raise over $755,000 for the foundation to help improve awareness, education, support, and research. From my personal and professional experience, I know him as a person of impeccable character. I find him to be incredibly passionate and compassionate towards other people, not only with Angelman syndrome, but in his dealings with those on the board and within our community. He has never shied away from asking the tough questions, but he's always done so in a very kind and caring manner. He has been an advisor, a mentor, a colleague, and a friend, and I know that I, as well as the Angelman Syndrome Foundation and the Angelman Syndrome community, are so much better for having him be such an influential part of this community and this organization. It is truly my honor and privilege to present the 2003 Harry and Audrey Angelman Award to this year's co-recipient, Mr. Davar Burbidge.
Wow. Is that really me? Thank you very much. It's a great honor to receive the Harry and Audrey Angelman Award. I'm also honored to be sharing it with Cindy Snyder, who will be following me shortly here, I'm sure. I'm very grateful to the ASF for this recognition, and I particularly want to thank the many board members and volunteers that I've worked with over the years. They've been a wonderful, dedicated group. Can everyone hear me okay? Okay. And in that group, I also want to include Eileen and Sheila, who have provided just wonderful leadership to the organization over these years. I would also like to thank my wife, Susie, who is here today. She is very understanding and supportive and allowing me to pursue my quest to be part of this Angelman army of conquest. We are in this room are about to hear how close we are getting to doing something about this condition that we called Angelman. Our kids are trusting us to make this happen. Please pardon me for being very blunt, but we need more soldiers in this battle, more volunteers. My 11 years as an ASF volunteer prompts me to issue this challenge to you. You parents are unbelievably busy with childcare. But what about your parents, the grandparents of these AS kids? I'm the age of your kids' grandparents, and I'm challenging you to set the grandparents loose on this Angelman disease. They may be hesitant to step into your lives in this indirect way, but you need to encourage them to do so. There are grandparents volunteering today in many of our Angelman walks across the country, including my city of Washington, D.C., but we need more across the country. The grandparents likely have more time to volunteer than you do. Please ask your parents to actively join you in this fight for their grandkids by becoming ASF volunteers. They can make a huge difference. So that is my challenge to you. Thank you again for this award, and let's go do great things for these kids.
Davar. All right. This year, we are, as I mentioned before, we're privileged to present the award to co-recipients. Unfortunately, our recipient is here, but she is not in the room. But I do want to continue on with the program this afternoon. The co-recipient of the Angelman and Harry, the co-recipient of the Harry and Audrey Angelman Award is a single mother to a now young man with Angelman syndrome. For many of us, we were thrust into the world of Angelman syndrome without a conscious choice or decision to be made. But this wonderful woman chose to adopt her son, knowing full well that he had Angelman syndrome. Not only did she not shy away from this, but she fully embraced the opportunity and then took her desire to care for and help other people with Angelman syndrome to additional dimensions. She's organized the Rocky Mountain Regional Angelman Syndrome Foundation Support Group. She's worked to bring workshops to the families in her area, as well as help and support other families with Angelman syndrome. But it doesn't stop there. Since 2005, she has organized one of our very successful walk sites, and under her direction and her team of volunteers, she has helped to raise, to date, $300,000 at the Denver walk site for the Angelman Syndrome Foundation to help improve awareness, education, support, and research on behalf of all of our children and loved ones with Angelman syndrome. So on her behalf, I would like to have you all recognize Miss Cindy Snyder from Denver as the co-recipient of this award. And with that, I'm going to turn the microphone over to Dr. Dan Harvey, the chair of our Scientific Advisory Committee. We're going to switch out a couple things here. You can have this just for this if you want this. This is a description of the work, okay? And then you can switch out.
All right. I'm on. Okay. So thank you very much. Can everybody hear me okay? No? Hello, just checking to see if everybody's awake. So it's a great pleasure for me to present this year's Claudia Benton Award for Research. I learned that I would be presenting this award a few minutes ago, so I apologize if my remarks are not as well prepared as Eileen. The criteria for the award are shown here. I want to talk a little bit before I present the award about Claudia Benton. Claudia was a scientist working in Art Baudet's lab in the 1990s. I had the pleasure of meeting her at my first Angelman syndrome conference when my son Matthew was, he would have been about 2 years old. Had been recently diagnosed. That was the first conference that I had the opportunity to attend, and met her at what I think was our first scientific symposium. Very warm, caring woman. Parents spoke very highly of her. I was very impressed by her, and she was a critical person involved in not only the identification of the ubiquitin ligase gene, the UBE3A gene that is associated with Angelman syndrome, as well as coordinating the activities at the Baylor, in Art Baudet's lab group at Baylor, and coordinating a lot of the interactions with patients that were going to that lab for guidance and support at that time. So she was really a tremendous person and leader who tragically was killed a few years or shortly thereafter that period. So a tremendous loss for the Angelman syndrome community, and this is why the Angelman syndrome Foundation decided to name its research award after Claudia a few years ago. This year's recipient, you're going to be hearing a lot about today. He became involved in Angelman syndrome research a few years ago. He was discussing some different things with some of his colleagues at the University of North Carolina, and that led to some studies of Angelman syndrome, and he's really, over the last few years, done a tremendous amount of very exciting, intriguing, and promising research, both to develop an understanding of the fundamental aspects of Angelman syndrome, but also to head us in the direction of treatments, as you'll be hearing about today. He also has been very, very supportive of the Angelman Syndrome Foundation, been very, very supportive in reaching out and working with parents, explaining the science of Angelman syndrome to the community and the broader audience, and helping to establish clinics associated with Angelman syndrome. One of them was in one of our clinics is now in North Carolina. So it's a great pleasure for me to present this year's Dr. Claudia Benton Award for Scientific Research to Ben Philpot. If you would come up here, please. I need to make sure I give him the right award. Congratulations. Let me hand this to you here. Do you want to say a few words now, or?
Sure.
Okay.
You're going to probably be hearing enough from me in a little bit, but I would say it's been an incredible odyssey for me to be here today, because it was not that long ago that I didn't even know what Angelman syndrome was. So what's something that's very scary to think about, that even after a pretty intensive undergraduate research that I was doing, in which I was doing neuroscience studies, after 5 years of graduate school and an embarrassingly long 6 years of postdoctoral research at Brown University and MIT, even after all those neuroscience studies, at the end of that I still didn't know what Angelman syndrome was. And it wasn't until a conversation with Mike Ehlers, who was then at Duke University, that he discussed a collaboration with Angelman syndrome. And at that time, not that long ago, it was just an intellectual curiosity for me and something to study from a scientific perspective. But I have to say, it's grown to be much more than that to me. And it's really through the interactions with the individuals with Angelman syndrome and the families that it's become something that's intensely personal and special. And it's brought an incredible amount of meaning to my research, and it gives me an incredible amount of motivation. So the community's been very generous to me, very wonderful, and I've just enjoyed it. It's probably a great passion of my life, and I thank you all for that.
So I think I'm supposed to just swap and move on. Where did I then go? Yes. Okay, give me 1 second here. Okay, if my computer wakes up here. All right, so what we're going to do is I'm going to provide an initial overview of Angelman syndrome, and I'm basically the warm-up act for Ben. Ben's going to go into a lot more detail about his work on Angelman syndrome. First of all, let me briefly introduce myself. I am a parent of a child with Angelman syndrome. A picture of my son Matt is shown here. This is a recent picture of Matt. He recently completed high school, and I think I'm very proud of the fact that we made it all the way from birth to this period. He's grown up a lot. Before you get too excited about this picture in your child, he has never worn a tuxedo. So this is a picture of him, but there's a lot of Photoshop involved by some very, very talented photographers that work for our public high school systems in the San Diego area. So I'm a parent, but I'm also a scientist. I'm an organic chemist by training. I have been monitoring Angelman syndrome research and involved in scientific activities of the foundation for a number of years. I did rotate off the scientific advisory committee a number of years ago and recently came back on. And am very excited about working with the foundation and the Angelman syndrome community once again to advance the scientific mission of the foundation and the community. So what I'm going to do here is try to relatively quickly go through some of the history of Angelman syndrome, and I want to start with the past. And for those of you that have been involved in Angelman syndrome for a number of years, there are going to be a few slides that are repetitive. But how many first-timers do we have at this meeting? Okay, so I do want to cover the background. It's very important that we recognize where we started from, really not that long ago, and understand how far we've gotten. But also then put that in the perspective of where what remains to be done, if you will. So really everything that we're dealing with started with Harry Angelman. You've heard a little bit about Harry and Audrey already. They were very involved in the foundation when at its inception, which I'll talk about in a second. But this all started with Harry and some observations that he made in the, I guess, early 1960s, which culminated in he was working with patients in England and identified 3 children that that had a common set of characteristics, and published a paper that was titled "Puppet Children: A Report on 3 Cases." And this publication was done in 1965, and relatively little attention was paid to it for a number of years afterwards. So this was the initial publication. And I did want to talk, before I go on, I want to talk a little bit about Harry. My source of, the source of all information now is Wikipedia. So I did go to Wikipedia and see what is posted now at Wikipedia. And, the background on this, and I think Harry, for those of you that have been involved in the foundation for a number of years, Harry had relayed this story to people, but he was traveled a lot and had visited Italy and had observed this picture at a art museum in Italy by Giovanni Francesco Carotto, and this reminded him of these children that he had observed, and it was this painting of a child with a puppet, and this led him to describe the syndrome as these puppet children as it was. As he viewed it. It's a very interesting paper to read. When my son was diagnosed, I did go back and review the literature in detail and did read the full manuscript. And it's very interesting to read this paper and put it in the context of what we know today. So that's where we were back in 1965. So think back to the 1960s and the 1950s and 1940s. Children with Angelman syndrome were being born at the same rate they are today, but they just had no diagnosis, and so there's a lot of background there that really the whole foundation of this started in 1965 with this publication. Relatively little was published about Angelman syndrome for a number of years following that. I originally had on this slide Charlie Williams, 1978 to 1982, which made it sound like Charlie only lived for about 4 years. But jumping forward to 1978, relatively little had been published. There were a few reports of additional cases of Angelman syndrome, but it was not recognized as being a very, very common disorder. So in 1978, and these are a quote again from the World Wide Web, these are Charlie's words here, which I have captured that he wrote at some point. But Charlie first became aware of Angelman syndrome in 1978 from work that he was doing with his teaching professor, Dr. Jamie Frias. They identified 6 cases, 6 individuals with Angelman syndrome. recognized that this might be a relatively common disorder because they had such a sizable pool of individuals with Angelman syndrome just in their local, in an institution in the local Gainesville area, and published a paper on it that was published in 1982. And shortly thereafter, the Angelman Syndrome Support Group that became the Angelman Syndrome Foundation was established with the leadership of Charlie and connecting with parents that were involved in some of those initial groups. And the publication that was done in 1982 is cited there. And Charlie is obviously here, and I think we all owe a tremendous debt of thanks both to Harry Angelman for his work in 1965 and Charlie's subsequent work and continuing involvement in Angelman syndrome and the foundation in all of our activities since then. So I have one slide here that is going to cover 14 years, and it's not that very little was happening. There was a lot of stuff happening during this period, but a lot of this is, things that are going to be covered elsewhere during presentations at the conference. So during this period, after Charlie's, the recognition by Charlie and then others that it was a much more common disorder than originally thought. A lot more cases were identified, so there were reports coming out, not just from Charlie's work, but from the work of others in the U.S. and abroad, that there were a lot of significant number of cases, and there were initial studies to try to determine the prevalence of Angelman syndrome. These were all just clinical diagnoses at the time. They didn't have any idea of what was going on with the genetics of the disorder. And as Charlie and others were they started to evaluate the genetics of these patients, and I'm not a geneticist, so I'm just going to oversimplify things, but they found that in many of the cases, these cases, they did identify that they had a sizable deletion on chromosome 15, and that deletion became associated with Angelman syndrome. Further studies identified a group of patients with a completely different disorder known as Prader-Willi syndrome that also had that same deletion, and this was very confusing initially, but it became I think really the key case that identified this concept of genomic imprinting, where we inherit some of our genes from our father. Some of our genes are specifically expressed from our father's genes. Our father's genes are specifically expressed in some cases, and the genes from our mother are expressed in other cases. And this was a whole new concept that was very intriguing, and came to light in the late 1980s or so. So that was a very new concept. When Matt was first diagnosed, I was reading in 1996, was reading about genetic imprinting. I went to my genetics textbook that I had used in a class that I had taken as an undergraduate, probably around 78 or 79. Tried to find out background information on genomic imprinting. There was absolutely nothing in there because it wasn't known at that point in time. Okay, so that's where we were. And I also then I was on the faculty at UCSD at the time, and I went to some of the textbooks that my colleagues that were teaching genetics taught. I went down to the bookstore and said, "What book are they using when they're teaching introductory genetics?" Wasn't in there as well. It just wasn't, had not been known long enough to have really been incorporated into most of the current textbooks at that point. They continued to evaluate, and they identified all the different number of different subclasses of Angelman syndrome, uniparental disomy, where both chromosomes are coming from the father. They identified another subclass with imprinting mutations, and a couple of other subclasses that I won't go into today. And these detailed, genetics. Charlie will actually be having a session on Genetics 101, and we'll be going into this in a lot more detail for anybody that would like to really get into this in more detail. There was still a sizable group of patients that had all the clinical symptoms of Angelman syndrome, but there was no obvious genetic cause that could be identified. Some of those cases were understood from reports that occurred in 1997. These reports came out of Art Baudet's lab at Baylor and Joe Wagstaff led lab, and he was at, I believe, at Mass General at the time. But both of these reports in early 1997 identified a single gene within that region of the chromosomal deletion as the gene specifically associated with Angelman syndrome. You will see it in the literature referred to as E6AP, and you also see it referred to as UBE3A. Different contexts, those terms are somewhat interchangeable as far as this discussion is concerned. Now, as a chemist, this was really an exciting development for me to know that we knew the gene. And it really, from my perspective at the time, this was when the Human Genome Project was going on, and a lot of excitement about genes and what we could learn from genes and develop disease treatments, that if once we have the gene, the general idea is we can understand the protein that gene codes for. Once we understand that, we can begin to rationally design therapeutics that might be targeting that gene product. At the time, I remember discussing it with people at meetings and on the listserv, which was very active at that point, which I was very active in at that point. And I was asked, how soon will this result in the development of therapeutics? And I said, well, probably 5 to 10 years. We'll know what the gene does, what it codes for, and then people like me can start designing compounds that might interact with that substrate. I was very wrong at that time. It's taken us a bit longer than that, as I'll explain in a few minutes. So that was a critical point. In 1997, the big question was, what does that ubiquitin ligase gene UBE3A do? This is a schematic of that process. If I had a laser pointer, Ben is right there. Okay, thank you. Okay, so this is a schematic diagram. This is basically a simplification of the ubiquitin ligase process. This is as much biochemistry. This is the most intense biochemistry I'm going to sort of walk through with you today. But the ubiquitin ligase pathway involves 3 key components, and they are labeled E1 here, E2 here, and then the one that we're most interested in is this E31 here. Okay, so ubiquitin UBE3A is a very specific ubiquitin ligase, and it's an E3 ubiquitin ligase. Okay, now ubiquitin is a small molecule or a small protein, and what this ubiquitin ligase does is it attaches through a chain of events. It attaches this ubiquitin to a substrate. This substrate here in green here, it attaches the ubiquitin molecule here through this nitrogen, and I'll skip all the details of that. But it attaches one ubiquitin, and then from there are subsequent pathways in which additional ubiquitins are attached. And back in 1997, the general idea was that this attachment of ubiquitin to a substrate was targeting, that was a way for the body to target that substrate, this substrate here, for degradation. It was, this substrate has done something, it's been used for something, the body no longer needs it, so it attaches this ubiquitin to it, and then this ubiquitin, there's this protein degradation machinery that then sort of eats up and chews up and recycles that substrate and does other stuff with it. It's not that simple a process. We know that this substrate, that this ubiquitin ligase pathway is much more complicated than that now. I won't go into the details of that, but this is sort of the snapshot that we had of it in 1997. The key question as far as Angelman syndrome is concerned is what exactly is this substrate, or what exactly was the substrate that this ubiquitin ligase is attaching to. Now, from the chemistry perspective, when we want to design a drug, a thing that we ask is a protein, is an enzyme, is something a druggable target? Can I, as a chemist, go in the lab, do I understand it well enough so that I can rationally design a small molecule that I can take or inject or do something with so that it gets into my system and interacts with that protein so that I can influence its activity? In the context of Angelman syndrome, from my perspective, others may agree or disagree with me, but from my perspective, ubiquitin ligase EBE3A in the context of Angelman syndrome is not a druggable target. Real simply, it's absent because it's not being produced. Okay, so I can't go in and influence something. I can't increase its activity, or I can't modify its activity because it's not there. Okay, so it itself is not a druggable target from the perspective of drug design and drug development. The thought at the time, at least going through my head, well, as soon as we figure out what this substrate is, this substrate, if it's being targeted for degradation, if I can understand what that substrate is doing, then I can go in and start designing molecules that might influence the behavior of that substrate and start developing treatments for that, for the disorder. So, and that's why I was saying in 1997, just a few years, molecular biologists will figure out what this substrate is, and we'll be off and running on treatments. That's where it gets complicated. So what was hoped was that UBE3A would just have one substrate, and I've identified that as protein 1. Okay, so the question was, is it just one substrate, or is it interacting and attaching this ubiquitin to several substrates? So maybe there's not just one, but maybe it's attaching ubiquitin to protein number 1, or protein number 2, or maybe protein number 3, or 4, 5, 6, 7, 8, 9, 10. Maybe there's a whole lot. So the question is there one substrate or several substrates, or are there a whole lot of substrates? A number of studies have been done of this issue over the last decade or so, and a lot of interest in sort of the last 5 or 6 years. And there's a number of groups that have been trying to identify these substrates, and the bottom line is that right now it appears that there are multiple substrates. And just yesterday at the meeting, John Hubretsky from the University of Texas at Austin, he talked about some of his studies where he was able to trap some of these proteins that are interacting with UBE3A, and indicated that there are, in his studies, which are somewhat preliminary but appear to be very solid, that there are at least 13 different substrates. Okay? So again, from my perspective on the drug design, that means I've got not just one target, I've got to come up with 13 different drugs if I'm going to go through the rational drug design process and try to influence these substrates. On the other hand, maybe 12 of these have little or nothing to do with the clinical presentation of Angelman syndrome. Maybe 2 or 3 of them do. So maybe we need to go after all of them, or maybe we just need to go after one or two of them. Okay? So that's kind of where we are as far as ubiquitination pathways are concerned. Now, there are this goes on. Once we start thinking about protein 1, protein 2, and protein 3, maybe these are druggable targets. Maybe we can start addressing therapeutic interventions based on, maybe start thinking about therapeutic interventions based on those targets. But maybe those proteins interact with other proteins. And this is where we start getting this whole protein, or whole family of what we refer to as biochemical pathways, where one protein interacts with another, which interacts with another. So maybe this protein interacts with several different proteins. So maybe proteins 4, 5, and 6 here are possible targets for therapeutic intervention. Maybe this protein interacts with 3 other proteins, 7, 8, and 9, and maybe these are targets for therapeutic intervention. And this could go on, and it could go out several steps, and maybe if we understand, eventually, once we do understand what UBE3 attaches ubiquitin to and what that interacts with and what that interacts with, then we can come up with a druggable target, and maybe we can start designing molecules for that. Okay, so this is a very sort of simplified version. Now, I apologize for this next slide, but this is the way that people like Ben and a few others in the audience start putting all these different pathways together when they're starting to analyze these kinds of pathways. Now, I don't think you're going to find UBE3A anywhere in here, but for the neuroscience company that I work with, this includes a lot of the pathways that me and my colleagues deal with on a daily basis. So each of these little colored schemes, shows one of those proteins. Some of these arrows here show how those proteins interact. This sort of, here, this is the nucleus. This is the cytoplasm. This is the junction between two different cells. So if I've got a drug and I want to go in and I need to, if I want to interact, if I'm, now actually one thing in here is the MECP2 gene that's associated with Rett syndrome. That is shown right here. If I want to influence that, I've got to get a drug in here and get it interacting with MECP2, okay? So that's the kind of thing that we worry about. And again, a lot more detail here, but that's how this, I think, gives you an idea of you know, how complicated and how quickly this can get very complicated as far as the molecular biology or biochemistry is concerned. So let me get that slide off the screen. So getting back to the development of treatments, there are generally 3 different approaches to treatments that we are pursuing with respect, or have been pursuing with respect to Angelman syndrome. The first is just treatments targeting the symptoms. Okay, so we know that children with individuals with Angelman syndrome have issues with epilepsy. So let's develop treatments specifically dealing with epilepsy. We know they don't sleep, have issues with sleep. Talk a few minutes ago indicated that approximately 85% of Angelman syndrome individuals have sleep cycle disorder issues. So that's clearly a critical issue. So let's develop treatments for that. So that is one thing that has been going on for many years. Clinicians and scientists have been looking at Angelman syndrome and developing treatments for specific aspects of the disorder. I'll come back to that in a minute. Another aspect of what I've been talking about in the last few slides is what we are been doing extensively since the identification of the gene in 1997 is studying the role of UBE3A from my perspective, the primary interest being in identifying druggable targets so that we can then start looking at those pathways and coming up with compounds that would interact with those targets and actually start address not just some of the symptoms, but the actual cognitive deficit potentially associated with Angelman syndrome, as well as some of the other clinical signs. The other third, which is, this is one that is particularly, sort of available because of the genetics of Angelman syndrome, is activation of the paternal gene. Okay, so in the case of Angelman syndrome, we have a maternal gene that is silenced, but we have a paternal gene that is there. Or I'm sorry, we have a maternal gene that is missing, okay, but we have a paternal gene that is there but silenced. So the question is, can we turn on in some way that paternal gene, okay? And that's, if we are ever going to come up with something that is actually going to treat the fundamental, that's going to actually serve as a fundamental treatment for the underlying condition of Angelman syndrome, it's activation of that paternal gene. That's probably our best bet to go in and address as many of the issues as possible. At this point, because of what we currently know about the role of UBE3A and the proteins or targets and substrates that UBA is associated with it, those may have, I would anticipate that treatments will come out from that. They will have an effect, but they will not address all of the underlying systems. They may help, but they might not completely reverse the phenotype or the clinical symptoms of Angelman syndrome. So with respect to symptomatic treatments, I'm going to defer to presentations that are going to occur later on in this meeting. Seizures are going to be discussed extensively. If you're having issues with seizures and you want to learn about treatments for seizures, Ron Thiebert and Mark Nisbecka, who are both members of our scientific advisory committee, are going to be giving presentations on that aspect. Sleep, there are presentations on sleep. And also, if anybody is tired, go ahead and take a nap. I don't mind. I'll understand if you didn't sleep well last night. Go ahead, take a break. You deserve it. But there are going to be discussions about sleep and managing and treating the sleep aspect of the disorder. Communication is a critical issue. We have a number of people: R.J. Cooper, Steve Calculator, Mary Louise Bertram, who some of you may have participated in a workshop that she gave yesterday, and Aaron Sheldon are going to be talking about communication at various different sessions at this meeting. And behavior is a critical part, and Brett Kuhn again, Jane Summers, and Logan Week, there's going to be a number of talks on behavior. And these are all cases where clinicians and scientists have been evaluating these symptoms of the disorder and developing best practices and treatments for these things over the years. Some of them aren't perfect. Some of them still need development. Some of them need continued exploration. But there is a lot that we already know and can do with respect to treating the symptoms of the disorder. The first thing that I wanted to talk to you, so I wanted to talk to you about some of the things that we are doing. Let me actually go back a couple slides. So I want to go back to this issue of UBE3A and the identification of druggable targets. Okay, so there's been a lot of studies of what are the substrates involved in interacting with UBE3A, and what are some of the downstream proteins from that. One of them that has been identified by a number of groups. Where am I here? There were studies reported several years ago coming out of Dr. Weaver's lab and Dr. Gersman's lab. They published a very interesting paper dealing with CAMK2, or calcium calmodulin-dependent protein kinase. This is an enzyme that is probably several steps downstream from the UBE3A protein, but it has been suggested that it has a central role in producing the developmental deficits in Angelman syndrome. And so the ASF is funding a study by John Listman at Brandeis University, who has a lot of experience in this calcium calmodulin kinase area, to specifically look at CAMK2 in the context of Angelman syndrome and help us to determine whether this is indeed a druggable target in connection with Angelman syndrome. So this is one case where we are targeting and trying to really find whether or not one of these downstream proteins, proteins downstream from UBA3, can be a potential target for exploration and development of treatments. Another protein that is being looked at is ARC. Now, ARC has been identified, and ARC stands for activity-regulated cytoskeleton-associated protein. It's one that has been associated directly with Angelman syndrome, so it is just one step from that ubiquitin ligase gene, UBE3A. But Jason Shepard, a relatively young scientist at the University of Utah, is going to be evaluating the role of ARC in Angelman syndrome, and those studies we are hoping will lead to an indication of whether that is a potential druggable target that we can use as an opportunity to try to treat Angelman syndrome. Another study. Now, this actually goes back a bit. We have identified, many. Majority of individuals with Angelman syndrome have deletions. Some of them have imprinting control issues. Some of them have UPD. A lot of them have, or a small percentage of them have specific mutations within the UBE3A gene. So another thing that we are still interested in doing, and Mark Zilka, we funded a study with Mark Zilka to look at those mutations in the UBE3A gene in specific detail, and try to identify hotspots in the gene, or in that protein, and really figure out exactly why those hotspots or mutations in that specific area of that protein trigger loss of function of the gene. So this actually addresses some of the still questions about that small group of patients that still just have a clinical diagnosis, but it also potentially, for a small subset, might have an impact on a clinical therapeutics as well downstream from that. I want to change subjects a little bit and talk about the development of drugs. Okay, and there's been a lot of discussion these days about how do we, now that if we have targets, how do we go about identifying drugs that interact with those targets? And there's been discussions on the listserv and Facebook and elsewhere about screening compounds, and I'm not sure if anybody here has been involved in those discussions, but a very common step when you have a new target one of the first things that we do when we're looking for a drug is to check and see whether any known compounds out there will interact with it, okay, and influence its activity. So rather than having to go in the lab and starting from scratch and designing a compound, let's just check and see if there's anything out there that will interact and have an impact. So a couple of examples of this that I thought our audience might be familiar with. Please do not run out and start treating your child with either one of these reagents. The first one is AZT. Has anybody heard of AZT before? Okay, so this was back when I was in graduate school. The HIV epidemic was just hitting the news, and the HIV virus was identified. So one of the first things they did, that we needed to get treatments produced, as quickly as possible. It was clearly a major issue. So they started screening collections of antiviral agents, and a compound that had been prepared in approximately 1964 at the National Institutes of Health, it had been explored at that point as a potential antiviral agent, was produced, studied, never moved into really clinical use at that point, but it was in a compound collection, so it was part of a screen for compounds active against this virus, this HIV virus, and was indeed the first treatment that came out to address HIV. Had a tremendous impact at prolonging the lifetime of individuals with AIDS and allowed for their therapeutic window to go on. And now, with more time, a lot of subsequent antiviral agents attacking HIV from a variety of different perspectives are available now. But the first step in going after that was a drug repurposing effort, looking at known drugs. Another example, and I realized after I got here that this is a great example, because if you look at your bags, you will see that Pfizer is prominently displayed on them. So sildenafil is the name of the compound. Anybody familiar with sildenafil? No one's shaking there. So this is more commonly known as Viagra. Anybody ever heard of Viagra? So Viagra was first being developed by Pfizer as a treatment for pulmonary arterial hypertension. It never went to the clinic or through clinical trials for that, although I think it was in clinical trials, but they realized fairly early on, once they got it into man, that there was a side effect, and that side effect was what ended up being a much more powerful market than the original therapeutic indication, so it is being used as a treatment for erectile dysfunction. Now, I learn things when I get ready for these presentations, but according to Wikipedia, there is another repurposing drug, there's another repurposing use for this. It can also be used to extend the shelf life of cut flowers. So if you have flowers that, the same, it's a PDE5 inhibitor, and the PDE5 pathways that in humans are associated with this are also associated in plants with cut flowers, and it slows the ripening of plants. I have no experience myself with either anyway. But that's what Wikipedia says, okay? Anyway, so this is just a little bit of background. Please don't take offense at any of these sidetracks I'm taking. Let's talk about repurposing as it is in the context of Angelman syndrome. So, basically this is what has been going on for a number of years now, of looking at known drugs, known compounds that can be potentially used as a treatment for Angelman syndrome. One of the first studies, and this was a study funded by the Angelman Syndrome Foundation I think funded around 1996 or so, published in 1999. Joe Wagstaff, who about that same time was involved in identifying the ubiquitin UBE3A gene, also undertook a small-scale clinical study with individuals with Angelman syndrome, exploring whether melatonin was an effective treatment for Angelman syndrome. So this is looking at actually a hormone commonly available, readily available out there on the market, whether it could be used. And this paper documented in a very nice way that, and I have it sort of bolded at the bottom here, a moderate increase in circulating melatonin levels significantly reduces motor activity during the sleep period in Angelman syndrome children and promotes sleep. And, again, at the conference today, a group from the Netherlands was reporting sleep studies of individuals with Angelman syndrome in the Netherlands, and I think it was 35% of individuals with Angelman syndrome are being treated with melatonin in their clinical group of of patients in Europe. So very commonly used, and this is a case where a drug, hormone in this case, is being used as a treatment. There was another trial that was run shortly a few years afterwards. That was the folate betaine trial. There was a hypothesis put forward that by introducing vitamins or nutrients that are involved in mediating methylation. Okay, so DNA or the DNA silencing is involved. There's methylation issues on the DNA, and if we could influence methylation, increase background methylation, that might have a positive influence on the disorder. So this folate trial was run. A number of investigators were involved. It was started in approximately 2003. There were actually 2 phases. The second phase involved not just folate and betaine, but also creatine and vitamin B12. So they tried 2 different versions of it. The conclusion after all of that work was that it was safe, but it was not effective in decreasing the severity of Angelman syndrome. Okay, so this was a study. My son was the first child enrolled in this study in San Diego. Others, there were a number of clinical sites, and Mark Nispeka, Lynn Berg were involved in San Diego. Wenhan Tan, who is here today, and Art Baudet was very involved in this. And basically the question was asked. We found that it was, and took a number of years, but we did find that it was not an effective treatment. A critical part of this, though, was that this was really sort of the first blinded, placebo-controlled type of trial that was done in Angelman syndrome. And so, even though the result wasn't what we might have hoped for, we learned a lot about how to do clinical trials, what were the issues. We definitely needed to have more careful assessment criteria. The bottom line is, if we had seen these kids all of a sudden talking and walking, and doing a whole lot of things, yes, we probably would have had clear-cut clinical evidence. But if we had only seen a small effect, our criterion measurement vehicles, the background information, the databases and things that we have about the clinical criteria for Angelman syndrome would probably not have been sensitive enough to pick up a small effect. So what was launched shortly thereafter, and basically driven by some of the results of that, some of the questions that were being raised when that initial study was done, was this AS natural history study. Now, anybody involved here in that Angelman syndrome natural history study? Fantastic. You guys are the people that I'm talking about here. It was started in 2006. There are currently 6 study sites across the country. Involves medical and developmental evaluations once a year. Currently, there are 283 participants enrolled. If you are not involved in the AS Natural History Study, I would encourage you to contact one of these centers and get involved. They are still enrolling patients. This is critical for the future of clinical trials. At the meeting the last couple of days, I was talking with a couple people from pharma organizations that were here to learn about the disorder, Angelman syndrome, and one of the things they mentioned is that they are very excited about working with, that they need this kind of information, information that comes out of this natural history study in order for them to get excited and know that the base of information is out there that they need to move forward with clinical research or preclinical work on Angelman syndrome. Another, again, focused on repurposing now. Another repurposing opportunity that is still actively being explored is an evaluation of levodopa as a possible treatment for Angelman syndrome. This was driven by a report in 2001 of 2 patients with Parkinsonism symptoms. So with some of these patients had a genetic diagnosis of Angelman syndrome, but were displaying some symptoms of Parkinsonism, and levodopa is a treatment used as a treatment for Parkinson's disease. And so these 2 individuals with Angelman were treated and did show positive effect. In 2001, this publication occurred. A number of parents worked with physicians to put their individuals with Angelman syndrome on levodopa, and so there were then among the Angelman syndrome community considerable anecdotal reports of improvement in abilities and the ability of children to learn from these cases of just parents directly working with physicians. And then there was some mouse data to support this, some preclinical data coming out of Ed Weber's lab. And there have been subsequently some other studies done of levodopa in the preclinical area. But a phase 1 study, a dose-finding study, was launched in 2009. That study was completed in 2010. And there is a phase 2 study that is still ongoing. It opened this phase was started in January 2011. They are still enrolling patients. Originally we were hoping to get to 100. They are currently at 65 and are seeking more patients to participate in this study. So if you are interested, I would encourage you to look into this and consider joining this trial. They still have open slots. It's very important that we get enough participants so that we have valid clinical data to report, to evaluate after this study is eventually complete. And Jennifer is the person to contact at Children's Hospital, or at Children's at Harvard. Anyway, so that's an ongoing study. And again, this is a case where we're looking at levodopa and possibly repurposing it as a treatment for Angelman syndrome. Another trial that is ongoing that's again a repurposing study is a trial involving minocycline. This is a very interesting compound. It's an antibiotic. It's in this tetracycline class, and you can see tetracycline means that it has 4 rings here. It's a very common class of antibiotics, has been around for a long time, and this specific one is attracted a lot of attention recently in connection with a number of neurological disorders. MMP9 is thought to be the target of this in connection with its neuroprotective properties, and it's thought that it may have some involvement in reducing inflammation in the central nervous system. It's being pursued in connection with Angelman syndrome, but it's also, there are clinical studies going on with a lot of other disorders, Fragile X, Parkinson's, ALS, Huntington's disease, and many others. I did a quick look at the clinical trials website, and I think there were about 20 or so trials that were active or recently ongoing or recently closed in connection with studying minocycline. A small open-label trial of minocycline was launched in 2012. Ed Weber primarily involved, but working with a number of parents. 24 patients were enrolled, and we are eagerly awaiting the results of this study. The study, my understanding from interactions that I've had with Ed, is that the study is complete. They are analyzing the data and hope to report it in the fall of 2013, fall being sometime later this year. Now, I do want to mention that just at this meeting on, yesterday, Epe Elgersma, who is here today with us, did report some preliminary studies, preclinical work with Angelman syndrome mice evaluating minocycline, and I want just to sort of, I think Professor Elgersma is in the Netherlands. There's a lot of interest in Europe about also pursuing a study of minocycline, so he was looking to confirm whether in his hands whether this was worthwhile to take into the clinic. And so far, his studies with adult AS mice show that there is no improvement with treatment, which is in contrast to some of the preliminary preclinical data that has been reported by Ed Weber and his group. These are very, very preliminary results. Ed Weber's results have not yet been published, so as a scientist, I am not able to review either of these Ed studies in detail. So really, Dr. Elgersma's studies are ongoing. We're looking for the Weber lab to report their results very soon, and we're hoping that we will have those findings in the not-too-distant future. If they show a positive sign, if the preclinical results are positive, I'm in contact with clinical investigators and some of the same clinicians that have been involved in other studies of Angelman syndrome. We are ready to launch a study of minocycline if the preclinical data that if we can confirm and validate the preclinical studies, and once we have the results from the open label trial that's been done, to further explore this. Okay, so that's an ongoing effort there. Another repurposing study, when the Angelman Syndrome Foundation did a round of, we had reviewed a whole host of proposals. A number of them were focused on repurposing activities, studying compounds, and seeing if those compounds would have an impact on Angelman syndrome. These are mostly directed towards preclinical work, trying to determine whether known compounds, known drugs will be active against Angelman syndrome. And this is one that we are moving forward with funding. Acamprosate was proposed to be studied by Craig Erickson at Cincinnati Children's Hospital. And the structure of it is here. And I am a chemist, so I've been including structures here. Maybe they don't mean much to you, but I can't live without a few structures in my presentation. But acamprosate has been explored. Where is it? It's a treatment for alcoholism, that's not in this abstract. I shortened this so it would fit on the slide. But he's going to basically be looking at this, doing preclinical studies, looking at its activity, and seeing if some of the biomarkers that are associated with Angelman syndrome are influenced by this disorder. And trying to come up with doing the preclinical studies that would justify, if successful, moving forward with this as a possible potential treatment for Angelman syndrome. Okay, so that's another one that is being actively pursued. And you're going to see a whole series of these probably over the next few years as we learn more about some of these other drugs and targets and how they might be going after influencing Angelman syndrome. Going back to that, so I've talked about treatment options, symptomatic treatments. I've talked about drugs and repurposing and trying to develop drugs to go after proteins that are substrates for UBE3A. That third possibility was unsilencing of the paternal chromosome. Okay, and there are a number of ways that are being considered to do that. One of them is a proposal coming out of Art Baudet's lab. And Art, again, he was involved in identifying the AS gene a number of years ago, back in 1997. He's been very active in Angelman syndrome research throughout the period since then. And Lin-Yan Ming is working with him on a study where we're using antisense oligonucleotides as potential therapies for Angelman syndrome. It's a very complicated process, and we'd be here till tomorrow if I was trying to explain this in detail, but this is a very promising approach. They are working in collaboration with ISIS Pharmaceuticals, a company that has tremendous expertise and experience with antisense technology that's based in Carlsbad. I know we have some parents that might be here from Carlsbad, so this might be in their backyard. But this is a very promising approach and one that is being funded and pursued, funded by the Angelman Syndrome and pursued on our behalf by Art Baudet and Lin-Yan Ming and their colleagues at Baylor. I've talked much too long. The future. So where are we in terms of what are we going to see in the coming years? There's going to be continued development of symptomatic treatments, and the Scientific Advisory Committee and the Angelman Syndrome Foundation are committed to continuing to develop treatments for the symptoms of Angelman syndrome. We are, as I've indicated, we are funding a number of studies that are in this general area of analyzing the UBE3A substrate and pathways and identifying druggable targets. And then eventually hoping to look at those druggable targets and come up with compounds that will influence their behavior. Then we're also very actively involved in supporting efforts to activate the paternal gene, what I mentioned with our Baudet studies, and also some of the work that Ben is now going to talk about from his lab are also falling into this area. Okay, and I think that's where I'm going to conclude. At this point, I'm going to turn it over to Ben and let the fun begin here. Now, I should say we are, as Ben comes up and we swap computers, once we are going to have questions take questions from the audience. There is going to be a panel session, and we're going to answer everybody's questions shortly. Okay? Thank you.
Get the lights down a little bit in the front so you can see some of these slides. All right, can everyone hear me okay? It's really a great pleasure to be here and to give you a little bit of update of our research. And one of my goals was to tell you about our research and also help you understand why we're doing it, and both the promise and limitations of some of our approaches. Now, I want to state very clearly so that everyone understands that all our research, be it basic or preclinical, we have one goal in mind, and that's to improve the lives of individuals with Angelman syndrome. So we take a couple of different paths to do this, though, and I've kind of divided my lab up into these two different paths. And one of the paths towards therapeutics is what I think you as parents are most familiar with, and that the foundations tend to typically fund, and that is to do drug discovery. And I wanted you to appreciate that there are both advantages and disadvantages of doing drug discovery to develop therapeutics. The advantage is it has the potential to be relatively rapid, in that we might be able to identify a compound that could relatively rapidly be translated into a clinical use. And that, of course, is the hope of what all of us would like. But there are also some disadvantages to this approach, and that's that it tends to be risky. Okay, so there are risks both in that you tend to be taking a gamble and trying something that might work. There are also risks associated in that you can spend, as I'll tell you, you can spend a lot of money, and the outcome is uncertain. So the risk is in that roughly 1 in 10,000 compounds that is tried in drug discovery makes it. Okay, so the success rate is 1 in 10,000 roughly. So there's a huge chance for failure when you're starting at a very early stage. Okay? As I'll explain to you later, it's extremely costly to do drug discovery and to bring something from an idea, from kind of an idea at the bench level scientifically to a therapeutic use. And there's also a risk associated with the mechanisms of why a drug failed isn't studied, then you also don't gain much benefit from the study that was performed. So you can put millions of dollars into the research, and if it fails, if you don't investigate why it failed, then you don't have any useful information that can then be applied to the next study to try to develop a therapeutic. So these are some of the risks and disadvantages associated with drug discovery. Now, the other tact that I think the parents are less familiar with, but I wanted to try to educate you about why I think is important, is to perform very basic research into the pathophysiology of Angelman syndrome. And this has an advantage in that it tends to be a very steady approach that provides foundational knowledge. And this knowledge can help guide approaches that almost every other scientist can use into developing therapeutics. So it's kind of a sure and steady approach. Now, the obvious disadvantage is this tends to be a slower process, and it does not directly lead to a therapeutic itself. So whereas I think the parents are most excited about drug discovery, and this is the type of research that the parent foundations tend to fund, I wanted to try to tell you why I think basic research is another important avenue. And so I'm going to take just a few slides to talk about some of the basic research that my lab is doing and why I think is important. And then I'll focus the rest of the time on our drug discovery efforts. Now, what are the types of basic research that you can do? We're doing several types. I wanted to just tell you two of the studies that we're doing. One is to determine the expression pattern of UB3A in the brain across development. So where is UB3A expressed and when is it expressed in the brain? And if you look at a mouse model, I want to show, the other thing I want to do is try to show you some of the raw data that the scientists are getting so you can appreciate that. So this is raw data where I'm showing you the expression of UB3A in a mouse brain at 3 different ages, postnatal day 0, 7, or 28 in a mouse. And the green fluorescence that you see is UB3A expression. Okay, so this is the protein whose loss is responsible for Angelman syndrome. And postnatal day 0 is the time immediately after birth, and postnatal day 28 would be like a young adolescent, analogous to a young adolescent in humans. And I think you can easily appreciate that the expression pattern of UB3A changes dramatically over development. Okay? So here UB3A is expressed throughout lots of the process of these brain cells, and here it's spread out because UB3A is expressed preferentially, at least, in the nucleus. Okay? So if you look early, this is an expression pattern of UB3A. And if you look later in the development, this is an expression pattern of UB3A. Now, this is very, very basic research, but to scientists, it tells us something very important. So although not definitive, it suggests that the forms, what we call isoforms of UB3A, might change across development. You might not be able to readily appreciate why this is important from a therapeutic standpoint, but for a scientist, this is incredibly important because if you're doing a therapeutic approach, for example, that uses gene therapy, which you might be reinstating UB3A expression in the brain, you might want to know which isoform or which type of UB3A to express and at what time. Okay? So this is incredibly important information, and the knowledge gained from this can guide the rational development of therapeutics. It also suggests that perhaps the function of UB3A may change with age, and this is something that we can test, because if UB3A plays a different role at different stages of brain development, then we should know what that is, because once again, this is basic knowledge, but this knowledge can help guide the rational development of therapeutics. So I wanted you to appreciate just a couple of lines of basic research that my lab is taking, and I want to show you one other one. And that's to determine the consequences of UB3A loss on brain functions. So the basic idea is to take the mouse model of Angelman syndrome and to look how the loss of UB3A affects development of the brain. Once again, very basic research. And to tell you a little bit about this study, I have to educate you a little bit about how the brain works, how neurons in the brain function. So you know that your brain is made up of billions and billions of neurons, 2 examples of which are depicted here. So one neuron has a cell body, and then it projects information, like it emits information, on a specialized process known as an axon. Okay? this signal, this electrical signal that's then delivered, is delivered to the next neuron that it communicates with across a specialized structure known as a synapse. And this is where an electrical signal is transferred to a chemical signal through a neurotransmitter and then picked up as an electrical signal again on this dendrite of the next neuron, which acts as a receiver of that information. So you have the axon that acts as an emitter of information. It's sending the information. Synapse, which is a specialized site of communication between neurons, in which an electrical signal is changed briefly into a chemical signal. And then you have a receiver of information along the dendrites that once again changes this chemical signal back into an electrical signal. And it's through wiring of billions and billions of neurons like this that your brain can function and communicate. Now, it's important to understand how synaptic transmission works and how it can go awry with the loss of UB3A. So this is just a primer of synaptic transmission as it may occur from one neuron to another neuron. And in this example, it's important to realize that these little vesicles are neurotransmitters. So these are just chemicals that can be emitted. So when you see these ads for drug companies showing these chemicals and you're talking about a chemical imbalance in your brain that might contribute to your depression and how that can be fixed, this is what they're talking about, these neurotransmitters. Now, when there's electrical activity in a neuron, that is detected by receptors at the end of the neuron. Okay? These receptors, when activated, cause this neurotransmitter to be released into this gap, this synapse. The neurotransmitter then enters the synaptic area, and then the neurotransmitter can bind to receptors on the next neuron. These receptors are then activated, and then you get an electrical signal in the next neuron. Now, the process doesn't stop there, because this process requires that neurotransmitter is replenished, so then that the neuron can readily release neurotransmitter subsequently, because you always want your neurons to be primed to be active appropriately. So in this process, these vesicles that contain all the neurotransmitter are taken back up, and then they're coated with specialized structure. These structures are then uncoated, and neurotransmitter is filled back into the vesicles that allows these neurotransmitters to be released again. Okay. Now, this is the basic process that if it occurs, you'll have normal synaptic transmission in the brain. Now, one of the things that we noticed in Angelman syndrome, when we're just doing basic research, looking at a very, very high resolution of a synapse. So this is using a specialized structure, a specialized instrument known as an electron microscope to image synaptic connections and neurons that communicate with one another. When you image these structures at a high resolution, you can see an accumulation of structures that look like this, particularly in the Angelman syndrome brain, that you don't see in a neurotypical brain. Now, what this means is it suggests to a basic scientist that there's an incredible defect that needs to be overcome. So in this process of synaptic transmission that I just described to you in a neurotypical individual, it can be different in an individual with Angelman syndrome. So you can have neurotransmitter release that's normal at the synapse, can activate postsynaptic neuron, the one it's talking to, but then something changes when you're trying to replenish this neurotransmitter, in that these structures are getting coated in these clathrin-coated vesicles, but then they're not getting uncoated. So what happens is they're not getting filled back up with neurotransmitter. Eventually, you get an accumulation of these empty vesicles that don't have neurotransmitter. And there's a loss of vesicles that do have neurotransmitter. So this means that eventually you can have a loss of normal synaptic transmission, at least at a particular synapse type in the Angelman syndrome brain. So while this is very basic research, and we're looking into the pathophysiology of what goes awry in the absence of UB3A, I think it's extremely important because it gives us information, for example, about druggable targets. Perhaps we can find ways to help these vesicles get replenished or to enhance neurotransmission at these particular synapses to act as a therapeutic for Angelman syndrome. So I wanted to impart upon the parents and caregivers of individuals with Angelman syndrome that basic research, while it might be sometimes hard for you to appreciate or understand as a scientist, I feel it's extremely important because it can lead to the rational development of therapeutics. So I just wanted to give you a flavor for that. And after giving you a flavor of basic research, which about half of my lab at studying Angelman syndrome is doing, I wanted to talk about drug discovery efforts, which probably the other half of my lab is working on. Now, before I do so, I have to remind you about a little bit about the genetics of UB3A, the gene whose disruption causes Angelman syndrome. Now, almost every single gene in your body is represented by two copies. You have one copy that you inherit from your mom and one copy that you inherit from your dad. And all of these, most of these both copies are active. Okay? So both the copy you inherit from your dad and your mom is active. Now, in the case of UB3A, it's very specialized in that only the copy you inherit from your mother is active. Okay? The copy that's inherited from your father is not active, it's dormant. Okay? So it's silent. So this is shown here in that the copy of UB3A that is inherited maternally from the mom is active. The copy that you inherit paternally from the dad is inactive because it's silenced through a process that's kind of beyond genetics. It's called epigenetic process. Okay? So although it's intact, the genetic information is there from dad's copy, it's just not expressed. So this is shown in the mouse model here. So these is a depiction of a mouse model of Angelman syndrome. So this is a section of a mouse brain that's kind of taken from ear to ear in a mouse, in which, once again, UB3A protein expression is shown in green. Okay? And this would be in a neurotypical mouse, in which you have expression of UB3A off the gene copy that's inherited from the mom, but you don't have expression off the dad's copy. So all of this expression of UB3A in the brain is coming from the mom's copy. Now, we can basically manipulate the genetic material in a mouse to be able to study it. And in this case, in a mouse that was developed by Art Baudette's group, there's a deletion of the maternal copy of UB3A. And there's actually, you can't see it here, but this is a slice of brain, and we're looking at UB3A expression in a mouse brain in which only the maternal copy has been deleted, and you can see that there's no UB3A expression. Okay? So a deletion of the maternal copy of UB3A is sufficient to get rid of UB3A protein in the brain. So this is really key to the biology of Angelman syndrome. So with this biology in mind, we took on a very ambitious drug discovery program to try to develop therapeutics for Angelman syndrome. And I should state very clearly that this is a project that went well beyond my expertise, and I enlisted help from a large number of other investigators. Notably amongst those are Mark Zilka, who is a neurogeneticist and also has expertise in molecular biology and biochemistry, and Brian Roth, who's in the Department of Pharmacology at UNC. And Brian Roth is a renowned expert in drug discovery. In fact, he is heading up the National Institute of Mental Health's psychoactive drug screening program. So he's one of these rare people in academics who has a pharmaceutical level ability to perform drug discovery studies. So I really tapped into their expertise for this project that went well beyond my own. Now, to understand the drug discovery process, I'll go back to the biology of Angelman syndrome. I'll remind you, in neurotypical individuals, UB3A is expressed off the maternal copy, so you have a protein product that's expressed. There's nothing expressed off the paternal copy. In Angelman syndrome, a mutation or deletion is sufficient to get rid of activity of the maternal copy, and since the paternal copy, the one from dad, is inactive, there's no UB3A expression. So a therapeutic strategy, then, is to find a drug that could turn on this dad's copy of the gene and replace the lost maternal expression with expression off the paternal allele. Okay? So this is the basic drug discovery approach. I want to point out that the initial funding for these projects came from the Angelman Syndrome Foundation, and then that initial funding also blossomed into funding from other organizations such as the Simons Foundation and NIH. So dollars coming from family members like yourselves really kind of grew exponentially, so it's something we're excited about. You should be applauding yourselves as the donors and supporters. So you can always count on an individual with Angelman syndrome for spontaneous claps. I love it. So it turns out that the tools to do this drug discovery project were already in hand. There was a mouse that was developed by Scott Dindot and Art Baudet's lab, in which he made a fluorescent reporter for activity of the UB3A gene. Okay? So in this mouse, into the maternal's copy of UB3A, they attached a yellow fluorescent protein. So this is just a fluorescent marker, which in subsequent slides will either show up as yellow or green, depending on how we enhanced it. And you can see in this section of brain, when the fluorescent reporter is on the mother's copy of the gene, you can see lots of neurons that are expressing UB3A. Okay? And we can tell this because the neurons are glowing. Now, if the fluorescent reporter is put on the paternal copy of UB3A, you don't see any fluorescence in the brain. Okay? So once again, this is just a fluorescent marker that through genetic trickery, we can either breed on to the mother's copy of UB3A or the father's copy. And it allows us to detect which gene is active, which gene copy is active. So this just shows you that the maternal copy of the gene is active and the paternal copy is inactive. Right? Now, we can then use this tool to do high-throughput drug screening. So we take mice in which there's the fluorescent reporters on the paternal copy. So these neurons do not have any fluorescence. We take neurons from these mice. We then plate them on 384 wells. So it's just a plate that has 384 little wells. We're filling up each well with approximately 20,000 neurons, all of which have the fluorescent reporter off the paternal UB3A. So there's no fluorescence. Now, the drug discovery part comes in, is now that we have these neurons plated out, we add drugs. So we add small molecule compounds. We're adding them in quadruplicate, so there are 4 sets of wells filled with the identical compound, and the readout of what we're looking for is 4 wells in which all the neurons are glowing. Because what this tells us is that we've activated the paternal copy of UB3A. Okay? So if we can do this, then this, once again, would represent a therapeutic opportunity. So I hate showing this next slide because what it really does is it cuts short about 2.5 years of intensive research from many research into one slide. But the next slide is going to show you all the compounds we screened and changes in fluorescence. So what you'll see on the y-axis is changes in paternal expression of UB3A, and on the x-axis will be the number of compounds we screened. Oh, before I do that, I forgot. I want you to appreciate what's required to do these types of high-throughput screening, because there are very few academic labs throughout the U.S. that have the ability to do high-throughput screening. And we're fortunate to have, once again, Brian Roth at the University of North Carolina, who has this kind of pharma-level drug discovery expertise and also this very kind of expensive $1 million equipment to do this. So what you're going to see is a short video here in which you're seeing some of the fluid handling robotics that manipulate all the drugs. So you can see that the computers, the computerized machinery can work much faster than an army of technicians ever could, and that they can move around drugs, add them to different wells, and allow us to read out whether there's activation of the paternal copy of UB3A. So I feel very fortunate, once again, to be at the University of North Carolina with this kind of level of expertise there and also this specialized equipment. Now, This is a slide I was talking about that cuts short years of very hard work. So this is paternal expression of UB3A, and these are 2,306 compounds that we tested, at least in our initial screen. Out of all of these compounds, there was only one called irinotecan that showed a significant increase in paternal UB3A expression. But this is exciting because this shows us that we have a compound that has the ability to turn on the paternal copy of UB3A. There's also another advantage. It turns out that this compound is known as a topoisomerase inhibitor, and it's an FDA-approved compound that's currently used to treat cancer. In fact, it's one of the most common cancer drugs that's out there is this class of topoisomerase inhibitors. Now, one of the things I learned about drug discovery is that once you have one compound that works, that gives you a great inroads to try to identify other compounds that may act similarly. So once we had one topoisomerase inhibitor that was able to activate the paternal copy, we were then able to test other topoisomerase inhibitors that have similar molecular structures. So this is just showing you another compound that works even better called topotecan. So this is looking at paternal UB3A expression when we just give a vehicle, just the saline solution that we use to inject the drug, so there's no drug here. When we add the topotecan drug, then we get expression of paternal UB3A. Okay? So it's very, very powerful activation of the paternal copy of UB3A. Now, you don't need to understand this slide here, but this slide is important for scientists because what it tells us is it tells us the dosing at which these compounds are effective. So this graph tells us that topotecan is much more effective than the original drug we found, which is irinotecan, and it acts at very, very low concentrations. And this is important because it means it might be a true clinical candidate, something that might be able to be taken forward. Now, it's one thing to show that these compounds can work in a culture dish, right? But what you want to really know is, can this work in an animal? So can it work in our mouse model of Angelman syndrome? So to do these studies, we performed intrathecal injections into mouse models that would report activation of paternal UB3A. Now, these intrathecal injections, first of all, I should tell you that it's a very standard and safe way of drug delivery. I'm sure a number of women here today have had epidurals during childbirth, and this is essentially the same route of drug delivery that you receive with an epidural. So why inject intrathecally? Well, these drugs, like topotecan, have been used intrathecally in patients to treat brain cancers. This is just an example of one study in which they've done that. And in this case, they did it in children safely. And so it could be an ideal route for treating individuals with Angelman syndrome. Now, the first step is to look at UB3A expression in the spinal cord and to make sure that just as in brain, there's a lack of paternal expression normally. So these are mice that have the fluorescent reporter, once again, paternally. This is a slice through the spinal cord in a mouse that hasn't been treated with drug, looking at paternal UB3A expression. You don't see any paternal UB3A expression there because there's no green. Okay? We know that there are lots of neurons there because this is a stain with a different marker called NuN, which just marks neurons. So all these blue cells are spinal cord neurons. So there are lots of neurons there, but they're not expressing paternal UB3A. Now, after we injected the mice for 2 weeks with the topoisomerase inhibitor, you can see that we've activated the paternal copy of UB3A in a subset of neurons. So just after 2 weeks of drug treatment, we've turned on the paternal copy of UB3A. Now, there are a number of limitations to our work in its current stage, and I want to, throughout this talk, try to explain these limitations to you. One of the limitations is that we're not getting the level of activation that we'd like in the spinal cord and in the brain with our drug treatments. So we can tell this by comparing what we're getting with drug to what we know we should get with maternal expression of UB3A. So we look at that. If we just use a different mouse that has a fluorescent reporter on the maternal copy of UB3A, you can see that there are a lot more neurons there that should be expressing UB3A that we're not getting. Okay? And this is a select example. So one of our limitations is that we're not getting the level of expression of UB3A throughout the brain as we'd like. Now, another question is the unsilencing of UB3A transient, or is it long-lasting? So this is important because if you give 2 weeks of drug, and if you were to wait a while after that, 2 weeks, and there's no UB3A activation, that would tell you that therapeutically you'd have to deliver the drugs continuously or at least relatively frequently at intervals throughout life, or at least throughout a critical period of development. If it were long-lasting, then you could give the drug acutely and then have long-lasting benefits because you wouldn't have to re-deliver the drug continuously. So it's a very important question as to whether UB3A activation by these drugs is transient or it's long-lasting. So to do this study, it's relatively simple. Once again, we turn to our mouse model. We inject the mice for two weeks intrathecally. Then we either look immediately after drug treatment, 4 weeks, 12 weeks, or 52 weeks after drug treatment. So there's no drug during this period. Okay? There's only drug during the first 2 weeks. I should also tell you for these studies, we dropped down the dosing of topotecan that we were given for a number of reasons. One is that we wanted to stay in a very, very safe range of drug dosing, and the other is if we gave low doses, then we get a sparse activation of the paternal copy of the gene and enable us to do automated counting of the number of cells that are expressing the paternal copy of UB3A. So the results are summarized down here. It might be a little bit hard to see with the lights, but this is in mice that are treated just without the drug, just with the vehicle that delivers the drug. There's no activation of paternal UB3A. Hopefully, you can appreciate that in drug-treated mice, there's activation of UB3A in a subset of neurons that can last out 52 weeks later. So basically, a year after we've given the drug, in a subset of spinal cord neurons, we get lasting unsilencing of UB3A. Just to blow up on that and highlight that fact is, once again, this is a section of spinal cord where we just gave a saline vehicle, and you don't see activation of paternal UB3A. This is a year after the last dose of topotecan, and you can see a number of neurons that have paternal expression of UB3A. So this suggests that a single dosing period of UB3A, at least in this subset of neurons, can have a lasting and perhaps a permanent unsilencing of UB3A. And that's incredibly exciting. Now, once again, I want you to appreciate a limitation that we also have, is that while we're seeing a permanent, or what appears to be a permanent unsilencing of paternal UB3A in these spinal cord neurons, we're not seeing that in other cell types, at least to date. So if we look at cultured neurons and we treat them and then take the drug away, very rapidly we lose expression of UB3A. So a very important avenue for us to study is why is it that in some cell types you're getting what appears to be a permanent unsilencing of UB3A, which would of course be the dream for a treatment, and in other cell types it appears to be transient. So if we can understand that, then it would make treatment with an acute administration of a drug that has known toxicities, right? I mean, everyone's watched someone go through chemotherapy. You know there are drugs that have toxicities that we need to appreciate and be aware of, because patient safety is, of course, our main goal. So if we can understand, from a basic research perspective, what is the switch that can turn UB3A on permanently, it's going to be incredibly important. Is it that we just need to get to the right dose in the cells that it's not a permanent switch in? Is it that there's something unique about the molecular composition, or what is it? So this is something that we're very actively interested in. Now, once again, you might be saying, Ben, it's It's great that you've been able to turn on the paternal copy of UB3A in a mouse, but we don't have mice, we have kids. Does it work in human neurons? Because we know that there are very important differences between a mouse and a human. And the question is, okay, you've got it to work in mouse, will it work in humans? So to do this study, we turned to a collaboration with Stormy Chamberlain at the University of Connecticut, who's really an amazing researcher, and hopefully someone that all of you get to hear talk. And what she has done, she has taken essentially skin cells from patients with Angelman syndrome. When you take these cells, there's now a specialized technique that you can, what we call, de-differentiate these cells so that they can become pluripotent stem cells. So they can be transformed into cells that have the potential to go into other cell types. So then she can transform these cells into neurons. So you can start with skin cells from an individual with Angelman syndrome. Through molecular biology, you can then force their fate to eventually go into that of a neuron. So from these skin cells, you can study then what it looks like to have a neuron from an Angelman syndrome patient. Then with these neurons, we can try these compounds. We can try topotecan in these cultured human neurons and see if we can activate the paternal copy of UB3A in human neurons, actually patient neurons, just as we did in mouse. And these are unpublished data showing you that, in fact, we can. So these are UB3A, paternal UB3A levels from Angelman syndrome patients. This is from a neurotypical individual, the kind of the level we'd like to get to. With increasing concentrations of topotecan, we get more and more expression of paternal UB3A. So just as in the mice, we've been able to activate the paternal copy of UB3A, we've been able to do so in the human neuron. So it's something that we're incredibly excited about. Once again, this is another point to interject some of the limitations we're facing. And I can tell you that the concentrations and the duration of which we have to keep on these drugs is higher in the human neurons than it is in the mouse neurons. And we think we have a handle of why this might be occurring, but it's important to realize that the dosing appears to be a little bit different in the human neurons than in the mouse neurons. Now, as I said, a theme that I want you to appreciate some of the current limitations and directions of our research. One is that topotecan does not unsilence UB3A throughout the brain. So we can even do studies where we infuse topotecan directly into the brain of mice. And what we'll see is we'll get great paternal activation around the site of infusion. So if we infuse it in an area known as the ventricles, we see great activation of an area of the brain known as the hippocampus, but we might not in a more distal region. So near the site of infusion, we get great unsilencing of paternal UB3A. As you move further and further away, you see less and less unsilencing. So this could really limit our ability to see behavioral recovery. So we need to identify additional topoisomerases and additional topoisomerase-like compounds that get into the brain better. Okay? Our current compounds, they either don't get into the brain that well, although they do get there. They can access the brain and cross the blood-brain barrier, but they don't do so particularly well. And even when we put them into the brain, they can be rapidly extruded from the brain. So we need compounds that can get into the brain and activate the paternal copy of UB3A. We also need to determine off-target effects that may limit chronic use of topoisomerase inhibitors. So you all know that acute treatment of these drugs is in the use as a chemotherapeutic has toxicities associated with them. Like I said, you've all probably had the misfortune of watching someone go through chemotherapy, and you know that it's an unpleasant experience and something that has side effects. So if this is something that we had to deliver over a long time, then one, we know already that there are side effects associated with that, and also if we're affecting other genes in addition to UB3A that we don't want to be affecting, this could limit the chronic use of UB3A. So a path forward might be if we only need to give a brief treatment of these topoisomerase inhibitors that could have a lasting activation of paternal UB3A, but not other genes that we don't want to be affecting. That could be useful. So a push of our research is to determine what other genes we're affecting, and is there a way to limit the activation of genes that we don't want to be activating. Right? We also need to determine the mechanism of action of these drugs to guide other unsilencing attempts. So if we can understand how these drugs are really unsilencing the paternal copy of UB3A, even if these drugs don't move forward themselves into the clinic, the knowledge we get from the ability to activate the paternal copy of UB3A could guide other attempts with safer, better drugs to unsilence the paternal copy of UB3A. So once again, this gets back to a basic science question where there's an opportunity to develop these compounds into therapeutics, but even if we don't, the knowledge we've gained from this is going to be able to guide other therapeutic attempts. And in fact, just at this conference that's going on now, there are at least 2 other investigators that are using topoisomerase inhibitors as a tool to understand the unsilencing mechanism. So that's very exciting for us. There's also a need to perform additional drug screens, both unbiased and targeted drug screens. So we're continuing to screen other compounds that might unsilence the paternal copy of UB3A. And of course, the dream is to have something that's as safe as aspirin, but as effective as these compounds here. So we're continuing to screen compounds, and we'll do so until our funds for that project run out. Now, we mentioned that we need to identify additional topoisomerase inhibitors. Along those lines, we formed a collaboration with Mark Cushman and Yves Pommier, which are some of the leading researchers in the world at developing chemotherapeutics. So they're developing topoisomerase inhibitors that might work better for different forms of cancer. But of course, they're eager to see if these compounds might also present an opportunity to treat Angelman syndrome. And perhaps some of these compounds might get into the brain better and have fewer off-target effects. So this is, once again, preliminary unpublished data where on the y-axis we're plotting the activation of paternal UB3A. So anything above this white line means we've activated it. Okay? This, as a reference, is our most effective dose of topotecan. And this is, all these are experimental compounds provided by Mark Cushman and Yves Pommier. And you can already see that we have at least 5 compounds that are as effective at topotecan. So these also present clinical candidates. So one of the things we want to do is to test these other compounds to see if some of those might get into the brain more effectively and have fewer off-target effects. Okay? So this is an exciting avenue of research. I can tell you that now we have approximately 30 different topoisomerase inhibitors, all of which can effectively unsilence the paternal copy of UB3A. We also think that topoisomerase represent a good molecular target for drug treatment in that it's expressed in the same cells that have UB3A. So where we're inactivating topoisomerases in the same cells where we want to turn on UB3A. So this makes it a good molecular target. Now, this video is to kind of show you the dream of what we're trying to do with this research. So this represents 2 neurons in which there's the silent paternal copy of UB3A, and then there's the mutated or deleted copy of UB3A. And the neurons aren't functioning exactly as they should because they lack UB3A expression. And the dream of what we'd hope to do is to find a drug treatment. Try this one more time. So you have a drug that would kind of fly in here like snowflakes and activate the silent copy of UB3A and then make the neuron happy and functioning properly. Okay? So that's the dream of what we're trying to do. Find these compounds that can activate the silenced copy of UB3A, restore function in neurons, and do so in a way that does not produce adverse side effects. Now, I've heard from a few parents, they've had a number of questions and concerns. So here's what I'm going to try to anticipate questions that you might have based on questions that I've heard from a few parents, and try to answer some of the pressing questions and concerns that you might have. And of course, anything that I don't answer up here on the stage, I will of course answer in the question and answer panel session that we're going to have following. But one concern is that why did you only screen a few thousand compounds? So some of you who are familiar with drug discovery know that a typical drug screen from a pharmaceutical company might screen millions of compounds to try to find a therapeutic, and we've only screened a few thousand. Well, you need to appreciate, first of all, that we have incredible expertise at this, so we're not doing this blindly. I'll remind you that Brian Roth, who's leading this part of the study, is the psychoactive drug screening, runs the psychoactive drug screening program for the National Institute of Mental Health, and he has one of the largest academic screening centers in the world. He also has access to very large drug libraries. So he has drug libraries of hundreds of thousands of compounds. So it's not that we don't have the drug libraries, we have huge drug libraries. But it's incredibly difficult to do a drug screen in neurons, as these are non-dividing cells, okay? So when you hear about a drug screen that's performed with millions of compounds that a pharmaceutical company has done, they've used dividing cells. So they can just get cells, they can put them in a dish, they can let them divide, and then they can form as many dishes as they want full of these cells that they can then screen with millions of compounds. Now, unfortunately for the drug discovery efforts in Angelman syndrome, the paternal copy of UB3A is only silent in brain neurons, and brain neurons are non-dividing cell types. So we cannot perform this screen in a dividing cell type. So every time we need to perform a screen, we unfortunately have to sacrifice a mouse and collect neurons from it. Okay? So we have in fact performed one of the only successful neuronal screens to date, and if you scour the literature, you'll see that there have been very, very few successful drug discovery screens performed in neurons, and our group has done one of the few. And we, kind of a back-of-the-envelope type of estimate, we estimate that we could have screened millions of compounds had we been, for the same kind of price and effort, had we been studying dividing cell types. Okay? So when a parent asks me, why do you screen just a few thousand compounds, this is an answer why. So to try to increase our chances of moving forward quickly, though, we focused our screen using the following guidelines. So we focused on FDA-approved compounds, neuroactive compounds, meaning compounds we know at least have some access to the brain, and or other drugs that we are suspected of being able to change the activation of genes. Okay? So all of these focus on these types of compounds would increase our ability to expedite something that we find from drug discovery into a therapeutic. Okay? So there are roughly 2,500 FDA-approved compounds, and we screened almost every single one of those. Okay? So we've screened almost the entire library of FDA-approved compounds, and we're going to continue to screen these compounds until our funds run out. Okay? So just to remind you, it's simply not economically feasible to screen large numbers of neurons in unbiased chemical matter, as it might require screening millions of compounds with the hopes of getting anything to clinical trials. And if you're doing this in these non-dividing cell types and neurons, it's just not feasible unless you have absolutely enormous funds. Now, I wanted to also let you know that the average cost of bringing unknown chemical matter to clinical use is enormous. So this is an estimate as of 2009 in a published manuscript by Bernard Munoz, showing that the average cost for a pharmaceutical company to bring a chemical matter from an idea to clinical use is $1.5 billion. Okay? So I wanted parents to kind of appreciate the enormity of what's required to bring something from an idea to clinical use. So another question that parents have asked is, have you tested whether topoisomerase inhibitors can treat the Angelman syndrome model mice? And if not, why not? Well, we haven't. And the reason we haven't is the current compounds we have only modest brain penetrance. Okay, so just only a small region of the brain exhibits unsilencing. So behavioral rescue is unlikely with our current compound. So we just don't think it's a worthwhile endeavor to start doing a behavioral rescue at this time because it's unlikely to succeed. Okay? So instead, we think our time is much better spent finding topoisomerase inhibitors with optimal brain penetrance and limited off-target effects. And this will increase our chance for a successful outcome. So instead of pursuing something that we deem as being likely to fail, we're instead placing our efforts into finding the best compound, whether it's a topoisomerase inhibitor or another compound, to bring forward that would be much more likely to succeed. Okay. Now, we also get asked the question of how likely it is that topoisomerase inhibitors will become a treatment for Angelman syndrome, and what is the time frame? This is a question that I absolutely hate. I don't like to answer because it's unanswerable. I would say that the answer would be sometime between a year and never, and that's an extremely unsatisfying answer, I'm sure, to a parent or caregiver, but at least in my opinion, that's the truth. We just don't have enough knowledge now to know if they could even ever make it as a therapeutic. And the last thing we would ever want to do is to bring something forward for a clinical trial if it weren't ready. Okay. I'll also remind you that it's kind of a long and winding road to go from an idea to a new medicine. These are just some images I lifted off the web showing you kind of what's involved in the pathway. And this is showing you that a drug discovery effort might begin with, let's say, 10,000 compounds, and 15 years later, they might have one compound that could be used in production as a treatment. Now, I'm sure hearing that it might take 15 years to go from a drug discovery effort to a therapeutic sounds like a really long time to you. And it is a long time, and it's an unsatisfyingly long time. So one of our approaches, as I mentioned and alluded to earlier, is to jump in later in this process and to try to speed up bringing something forward as a therapeutic. And one of the ways we can do that is by working with compounds that are already approved by the FDA. So an approach that my lab and other labs is taking is to let's first try compounds that are approved by the FDA that we know can be rapidly moved forward as a therapeutic and see if those work first. Okay? But at the same time, we know that these might not be the best approaches to go forward, so we're looking at other chemical matter as well and other treatment options. But I wanted parents to appreciate that I know everyone is ready for a therapeutic basically yesterday, that from a scientific perspective, I want to impart some kind of pragmatic realism on the parent part. It's a long process, and I assure you we're moving as fast as we can to try to move something forward quickly. But as I mentioned, we want to do so rationally and safely, because I think safety is a number one concern. Now, one of the reasons I'm optimistic for the future of Angelman syndrome therapeutics is the possibility to partner with pharmaceutical companies, because I really think that will accelerate and improve the quality of drug discovery. Okay? So it's one thing to have academic researchers trying to develop therapeutics for Angelman syndrome, but it also, of course, would help if we could get the might of pharmaceutical companies behind us and start to help us develop therapeutics. Because they, obviously, this is what they do. That's where a lot of expertise lies. And unfortunately, drug companies have traditionally stayed away from rare disorders because quite simply, it's not something that they can ever make money from. Okay, they can, at the end of the day, they have to pay for all the research that goes into it, and they can't spend, $50 billion or $1.5 billion on Angelman syndrome if the return is only going to be $100,000. So they really need to have profitability, and obviously, because Angelman syndrome is a relatively rare disorder, the opportunity for profitability is somewhat minimal. But things are starting to shift in the pharmaceutical industry because they're also realizing that rare disorders such as Angelman syndrome might be some of the most tractable disorders to try to develop therapeutics for. And the reason for this is that in these rare disorders, there's usually one gene that's causative of the disorder. So in the case of Angelman syndrome, we know the causative gene. We know that it's impairment of UB3A function. And from a scientific vantage and from a drug discovery vantage, this is incredibly useful and powerful. And in the case of Angelman syndrome, there's even more of a therapeutic opportunity because we know the paternal copy of UB3A lies intact, it's dormant, and we just need to find a way to awaken it. So pharmaceutical companies are starting to get engaged and look at Angelman syndrome therapeutics. And this is a great development and I think should be really encouraging to family members out there. And along these lines, I wanted to let you know that we have formalized our first partnership with a large pharmaceutical company. So we now have a formalized partnership with Genzyme, which is a Sanofi company, to do drug discovery efforts in Angelman syndrome. We're extremely excited about this partnership because they're a very well-respected company. And right now, the scale of the collaboration is quite small, but we hope that it will grow. And we're really excited by this. And as a sign of the dedication of Genzyme to this project, they had representatives from their company at the scientific meeting here, which I think is a really, really exciting piece of news for the families out there. All right, so as you know, we recently received funding from the Angelman Syndrome Foundation to study optimal treatment windows in Angelman syndrome model mice. And a question that I've gotten is, isn't this useless given that we do not yet have a treatment for humans, right? So we don't have a treatment in humans, so why would you need to study an optimal treatment in mice? Shouldn't we be placing our money in finding a therapeutic or advancing a therapeutic? Why do we want to look at an optimal window? And I'll tell you why I think this type of research is incredibly important. But to understand why it's important, you also need to understand why clinical drug trials fail. So you have to understand this. There's a lot of information on the web about this. This is just one website. But all you have to do is Google, why do clinical drug trials fail? And you'll get a wealth of information that's readily accessible and quite readable by a lay person. So many trials fail because of poor trial design. So the clinical trial is not properly designed or run, or the wrong clinical endpoints are selected. So we could take an extreme example. Let's say a clinical trial is run for an Angelman syndrome therapeutic. The endpoint that's chosen for this therapeutic is improved seizure resistance. So it's a seizure medication. It's thought to be a seizure medication. You give this compound in the clinical trial, and it turns out that it does nothing for seizures. But the individuals with Angelman syndrome are speaking at the end of the clinical trial. Okay? Guess what? That clinical trial failed. Okay? So it failed because they selected the wrong clinical endpoint. They didn't know what it might work and improve. So then the trial has to be completely rerun with a new patient population. And it's important to realize that the Angelman syndrome patient population for clinical trials is really quite limited, right? So it would have to be rerun at great expense, at great time, and with a new patient population. So it really would set things back. So why we're studying optimal treatment windows in Angelman syndrome model mice is, I think this information is going to be critical for guiding every single other preclinical study in mouse models as well as in human studies. So our studies will determine which behaviors recover during different treatment windows. So if you treat an Angelman syndrome mouse early in development, are you able to improve their seizure susceptibility? Are you able to improve motor cognition? If you treat them later in development, what can you improve? Once you have this knowledge, then you can rationally design future preclinical studies as well as clinical studies. So you can have the right endpoints for those studies. So although this research is not immediately moving for a treatment, I believe it's one of the single most important things that you can do, because it's going to guide rational studies for every other preclinical type of research or clinical study that's done afterwards. So you're much more likely to select the right endpoints in your study. Okay? So that's why we're doing that basic research. So the information is going to improve the outcomes for both preclinical and clinical trials, and this will increase the success rates for nearly all treatment approaches. So it's basic research, yet it's going to be widely applicable, and I think will really help speed forward preclinical and clinical trials that move forth after this study. Another concern that was raised to me recently is we noticed that you now have a grant to study Rett syndrome. And if you're studying Rett syndrome, does this mean your lab will be doing less Angelman syndrome research? Now, I can assure you that the Rett syndrome community was not upset that I started studying Rett syndrome, but the Angelman syndrome community, apparently there are a few members that were worried that I'm losing interest in Angelman syndrome. This simply could not be further from the truth. In fact, I had a focus on my lab that had been studying a particular type of receptor in the brain as a kind of a basic science study. That aspect of my lab has been phasing out, and it's been replaced with doing research on Rett. Now, the other thing is that in addition to this kind of being a lateral shift in studies, it's also going to be really beneficial for the Angelman syndrome community, because it turns out that there are a lot of similarities between Rett syndrome and Angelman syndrome. Although they're very, very distinct clinically, they still have a lot of overlap. And this is a review from Ron Thiebert where he pointed out some of the phenotypic similarities between Rett syndrome and Angelman syndrome, such as seizures, cognitive impairments, microcephaly, the lack of speech. All of these are phenotypic overlaps between Rett syndrome and Angelman syndrome. And by understanding Rett syndrome, we're hoping to get insights into Angelman syndrome and vice versa. I'm not alone in thinking that there's similarities between Angelman syndrome and Rett syndrome. Here are just a few studies from the literature showing that there seems to be some types of interactions between MECP2, which is a gene that causes Rett syndrome, and UB3A. So UB3A is involved in a subset of MECP2 functions, or I'm sure the converse is also true, obviously. So by shifting some basic research that I was doing on a particular type of receptor that wasn't relevant to a particular disorder, although it might be relevant to many disorders, to Rett syndrome, this is not going to take away from the Angelman syndrome research I'm doing in any way. In fact, it's going to enhance it, because we're particularly excited in looking at molecular and genetic overlap between Angelman syndrome and Rett syndrome and other phenotypically similar disorders, because we think this might give us other inroads into therapeutics by understanding the similarities between the molecular pathways that are affected in these disorders. So we think it's a really positive advance. All right. There's also concern that all Angelman syndrome researchers are working in a vacuum and that we don't communicate with one another. And I think a lot of parents are concerned that we're all working in silos and there's not collaboration between us. And I wanted to stress that, once again, this couldn't be further from the truth. This is a list of investigators just at the University of North Carolina with which we're collaborating. So these aren't people in my lab. These are entire other labs at the University of North Carolina with which I'm collaborating. So we're collaborating with Brian Roth on drug discovery. We're collaborating with Mark Zilka on looking at some of the molecular aspects of UB3A. We're collaborating with CJ Malenga and have a paper with him on looking at dopamine transmission in the Angelman syndrome model mice. Richard Weinberg, we're doing studies at the ultrastructure level, and the list goes on and on. Then you might ask, okay, are you collaborating with anyone outside of your university? And in fact, we're collaborating with a large number of people. So Ipel Gersma from the Netherlands and I are collaborating quite extensively to look at critical periods of development in Angelman syndrome, and we're sharing a lot of reagents. I'm also collaborating with investigators at Duke, with Stormy Chamberlain. We're working on her human neurons from Angelman syndrome patients. So there's a lot of collaboration between my lab, both with other labs at the University of North Carolina and also outside the University of North Carolina. And while it's true that I'm not collaborating with every other Angelman syndrome lab, this is actually a good thing because you want both collaboration, but you also want some parallel efforts. And a little bit of redundancy in science is actually a good thing because we can get independent validation from one another. What you don't want to see is one lab that keeps on telling you information. You want it to be repeated by other labs. So all of us want to cross-validate the main significant findings from one another. And through that type of built-in redundancy in some of the things we're doing, it actually is useful. So it's good both to have very, very healthy collaborations, but also to have a little bit of independence, because it also sparks creative approaches of going forward. If everyone's working together, we're all working on one approach. If there are many independent minds that are working forward, then there can be some novel, kind of orthogonal approaches to developing therapeutics. Okay? So I wanted to end on a note of optimism, because I think despite the caveats that I presented tonight, there's a lot of reasons to at least be cautiously optimistic for the treatment of Angelman syndrome. So we've shown you that UBE3A can be unsilenced in vivo. So at least in some cell types, it can be long-lasting, if not permanent. We also show you that topoisomerase can unsilence UBE3A in patient-derived neurons. And these compounds are clinical candidates for the treatment of Angelman syndrome. I would also say even if these compounds do not make it forward into the clinic due to the limitations I've described today, they're at the very least providing one, a powerful proof of principle that is encouraging me and other labs to move forward to find other approaches to unsilence the paternal copy of UBE3A. And they're also providing a powerful research tool to help us develop knowledge into how this mechanism of unsilencing is occurring, so we can use this to develop therapeutics. So other compounds or approaches may be able to unsilence paternal UBE3A. And I think through increased basic science discoveries, increased AS research, a pipeline of therapeutics that are now going on, and engagement of large pharmaceutical companies holds an incredible promise for the future of AS treatment. So I hope that all of you feel cautiously optimistic about the future, because I think there's a lot of reasons to be optimistic that therapeutics will be developed. I want to end by thanking very briefly my lab members and my collaborators, because they're really the ones that are driving all this. And I'm incredibly lucky and appreciative to my lab members. I have a great lab, many of which are sitting back there, and they're really wonderful. You know, and in fact, I think I'm going to embarrass them and make them stand up. So come on, Brandon, Matt, Janet, Kelly, come on, stand up. Portia's over there. So they're a wonderful group, and they're a lot of fun to work with. And as I mentioned, I get a lot of enthusiasm from the parents to do research, but I also get a lot of energy from my lab members. And of course, I want to thank my funding. Much of what I talked about today was funded by the Angelman Syndrome Foundation, and also in grants I got from that kind of seed money from the Angelman Syndrome Foundation that helped me get grants from Simon's Foundation and the National Institute of Mental Health for this research. So thank them, and thank you for your time and attention.
Those questions I think we have already addressed, so I'm going to go through some. We're going to go through some of those quickly. But if Wenhan and others, Jane, if you could come on up and I'm gonna, I think, stay up. I'm gonna stay up here, but we don't have enough room on the stage up here. And I've participated in these kinds of question and answer things before. I want to encourage everybody to keep your questions general. Often we end up with parents asking questions specific to their child, which is very difficult. I'm not a clinician. Many of the scientists here are, but many of them are not. And questions about your specific child are often best addressed by discussions here at the conference with clinicians where you can go into detail. So please keep your questions general. So I think we have one question from the audience. Hold on. We're going to turn on your mic. I think you're ready.
Okay. I'm Grace. I'm from Las Vegas. And my question is, it's a general question, and it's more out of curiosity. Anyone can answer the question. I am just wondering, out of all the enumerated list of names of researchers that the last speaker mentioned, that there is a collaboration amongst all of you. Why is there not a mention about Dr. Weber collaborating research with Angelman syndrome? And I know he's done research with Angelman syndrome. So
I'll pass it off to the panel in a second. So in my talk, the Angelman Syndrome Foundation has funded a number of studies by Ed and his group in the past. I am communicating with him about some of the ongoing activities of minocycline. He has worked with Dr. Alguerzma and is collaborating with many groups. So I'll defer to maybe Dr. Alguerzma. You want to comment on that, since you?
Well, is this on? No. Our group has really extensively collaborated with the group of Ed Weber. In fact, this is how we both really started working on the Angelman project. I think in the last few years, our approaches have differed, whereas Weber really has sought to go for approaches that would lead to something maybe very quick. We have been taking a more conservative, maybe more European approach, where we really emphasized that whatever we do, we have to learn from the steps that we do. And so we have invested a lot in going slowly and taking step by step. But we are actually in contact. And in fact, our lab has done extensive studies now on the minocycline project. Unfortunately, most of these studies were negative, but we will be in touch with Ed Weber very soon and put the data side by side and see how we can have come to such a different conclusion.
And I should add, Ed was not at the meeting this week, but many of his members of his lab were, and were at the meeting and participating in the discussion and dialogue. So,
yeah, I think it's good that you say that, because I spent at least 2 or 3 hours talking to all the members of the Weber laboratory. So.
Okay. More questions from the audience, or I have questions from the rest of the world. Yes, Davar.
Maybe I can make myself heard. Give me a frame of reference. How common or uncommon is it to develop therapies for genetic disorders of the nature we're working on? Is that commonly done or not?
I can comment on that, because we are actually working on a number of developmental disorders, and we have currently several large trials going on, randomized controlled trials, and we're definitely not the only ones. I think Angelman is lacking behind, because we need still to understand a lot of the basic science. There's a lot of questions still that we don't have addressed. For other trials, we are much further, because we understand some of the mechanisms, and we have drugs, and some of these drugs are already approved to be used. So it really depends on the disorder and what we know about the disorder. So the more we know about the mechanism, the more trials we have running. and the better the drugs are, also in the preclinical models. But of course, when in disorders where we know less, I think still has to be done more on the preclinical side before we can really do large clinical trials. But so the basic answer is there's a lot of clinical trials going on right now with a number of disorders.
Yeah, I would point out that there are other disorders of similar prevalence, such as Fragile X and Rett syndrome, for which there are trials that are ongoing that are kind of further along, once again, than in Angelman syndrome. It also might be a useful case study to look at some of these trials, such as Baclofen for Fragile X, which is a trial that recently ended because the researchers felt that they had chosen the wrong clinical endpoint. So that's something that you can Google and look at, because it's something that was showing some promise, yet the wrong clinical endpoint was chosen, and they essentially ran out of money then to redo the project. So it's something that there's a lot of talk about in the Fragile X community right now about. So it's something maybe we could learn from studying that.
Okay, I'm going to maybe direct the panel to some of the questions here, and these were collected, and we received the questions last night. I have not edited or screened them in any way. I've just gotten them out there, so some of them may not make a whole lot of sense to me, but maybe some of the investigators. So, and these are in no particular order. I have not had the opportunity to group them. So the first one, if I did not find evidence of Angelman, trying eczema test can help finding it. I was hoping to have Charlie. I think this has to do with genetic testing. So Wenhan looks like he might have something to say.
Okay, so I think Charlie would agree with me, but basically, personally, I do not believe in a pure clinical diagnosis of Angelman syndrome. I think notwithstanding what's written in textbooks and journal articles, fewer than 1 or 2% of individuals with Angelman syndrome do not have an identifiable molecular finding. Most of those individuals who are thought to have Angelman syndrome but have been tested negative probably have a different genetic disorder. My suggestion is that those individuals should be fully evaluated by a clinical geneticist, and depending on what we find on clinical evaluation, we may perform other tests, of which exome sequencing is one, but by no means the only clinical test out there.
Okay. So the next question was directed towards Ben, and this had to do with HDAC inhibitors, and sort of a bit rambling here, but have you, Ben, can you comment on this question or series of questions?
Yeah, so the question is, have we used HDAC inhibitors to see if they might cause unsilencing or enhance unsilencing of the paternal copy of UB3A, I think is what the question is getting at. I can tell you that where our research started was doing screens using compounds such as HDAC inhibitors to try to activate the paternal copy of UB3A. And all of those studies yielded negative results, and in fact, some of them actually decreased expression of UB3A. So it wasn't until we performed the unbiased screen that we found that topoisomerase inhibitors were effective. So we don't think that HDAC inhibitors are something that we're likely to use in combination with topoisomerase inhibitors to enhance that activity.
So the next one had to do with severity of the disorder with respect to genetic subclasses. Some of those parents are observing that deletion-positive kids seem to have more severe disabilities than UPD or UBE3A kids. Anybody on the panel want to comment on that?
So yes, so in general that is true. I think children with who are deletion positive tend to be more severely affected than those with UBE3A point mutations.
If I can maybe expand on that, I think the important thing to recognize is that the key clinical aspects of the disorder are present in all classes. Okay, so there are some differences, and you will see this in some of the kids out here, that there is a very wide range. And the range of abilities in those classes does overlap, and so there are kids that are relatively high functioning that are deletion positive, but generally the range of abilities does is quite wide in all the classes. This was a specific question concerning deterioration of skills. 29-year-old, their daughter is 29 years old, and there's a degradation in skills, so less walking, and the basic question, if I can abbreviate this, is this commonly seen as individuals age? Is there a reduction in skills? Wen-Han, maybe from the natural history study, do you have anything to add on that?
Right, so we don't really have enough adult participants in the natural history study right now to be able to answer this question definitively. There's some suggestion that I think their motor skills, particularly in terms of walking, in some of the kids becomes worse. Whether that's because of increasing muscle rigidity that you see in some of them, or whether it's because of worsening scoliosis remains to be determined. But yes, I think that's an anecdotal observation that some of us have seen.
I think there are also some studies. Joe Clayton Smith, in particular, in Europe, I think, published a study a number of years ago of adults with Angelman syndrome and did observe some of these types of findings that mobility was one of the key issues that they were seeing some decreasing mobility in adults. Very important to include physical activities and appropriate. It's a justification for having therapeutic interventions continuing on into adulthood to make sure that there's no loss of skills.
Can I just make a pitch on this, that this is precisely why we really need your help in coming to enroll in the natural history study, especially if you have older children and adults with Angelman syndrome. We really would love to see them. So if anyone is interested in this, please come and look for me afterwards.
So I think this next question, my grandson is experiencing issues, but the question here is we now have clinics in a number of locations, and the question is which clinic should they go to, Boston or Carolina? Now, Wen-Han, being from Cambridge, is very familiar with the Boston clinic. Ben, being from North Carolina, is very familiar with the North Carolina clinic. So I'm guessing that we're going to have a recommendation, whichever one you want to. I think they're both doing great work. But any care to comment on this?
I think it all depends on where you live. I don't think it really matters whether you go to North Carolina or to Mass General. And I would point out that the clinic in Boston is at Mass General, run by Dr. Ron Philpot. It is not at Boston Children's Hospital. We are two different institutions.
And I should add, one thing that Ben did not mention is that he has a research group at UNC. I just learned today that some of his scientists that are doing non-patient oriented work are actually participating in the clinic there and working with the patients on a number of studies. So it's very exciting, I think, to see the bridge that is being made between the neuroscience research and the clinical work that's being done at North Carolina. This last one saw this program on NOVA a couple of years ago, a bit lengthy but amazing. They use one's own DNA library to cure the disease. Why can't we use this method to treat AS? Please watch it. It is very revolutionary. If I had time, I would have checked this video out. I have no idea what this video presents. Have any of you watched NOVA for the last several years? I think we may just have to defer comment on that one. We have a question from the audience, so we'll go there.
Yeah, thanks for letting me jump in. Since we have scientists from both here and Europe, I was wondering if each could comment on how it is to deal with the regulatory body that you usually deal with, and the differences between the two, because that might inform where we want to do clinical trials in the future.
Well, I would say
FDA and the European counterpart.
I'd say that the regulations are extensive both in Europe and in the U.S. at the moment, which
so
is
equally bad or equally good?
Well, it depends on your perspective. I mean, I think good. They're from our point of view, they're probably arduous, both for working with mice and the protocols we need to work with mice, and the same is true for even more so if you're starting to work with humans. There's a lot of regulations, and it's a good thing. It's they're designed to protect the mice. They're designed to to protect the patients. So although there are extensive regulations, at the end of the day, it's a good thing.
Yeah, I think I tend to agree. It's harder to work with animals in Europe, that I noticed, because I worked with animals here in the United States. A lot harder, I would say. I think for children, working with children is going to be hard wherever you do it, and this is for a very, very good reason. So I don't think any one of us would like to see that any different. I think cost effective, I've seen several trials that we run. I've seen the counterparts in the United States. I think the costs are a big difference because we don't have the high part of the insurance, and the doctors and the medical care is a lot cheaper. So in that respect, Europe might be slightly better. But
Europe might be slightly better in terms of cost, is that what you said?
yeah.
Okay. Thank you.
I'll go back to the screen here. Next question is, what news updates are there on the research for curative therapy for SA? This is probably somebody from Europe. Syndrome de Angelman syndrome, so that's probably what's going on. I think we've provided excellent updates, so I think we'll pass on that one. Are any other compounds other than topoisomerase inhibitors successful in activating paternal gene to a lesser degree? Ben, this is to you.
So far, although we have about 30 compounds that activate paternal UV3A, all of them are topoisomerase inhibitors. We, at the moment, don't have another compound, at least it's validated, that can activate the paternal copy of UV3A. But as I mentioned, we're still doing the drug discovery effort, and we still have data to analyze, so we're hoping that we might get something in the remaining drug screen.
The next question Ben did address during his talk. If current candidates for therapy are feasible, will the therapy be lifelong, or does it activate the quiet chromosome for good? This has to do with the unsilencing approaches. So Ben, you want to reiterate? Or
just to reiterate, although we appear, at least in the mice, to get what appears to be permanent unsilencing in a subset of spinal cord neurons, in other neuron types, we're getting only transient unsilencing, and we don't know whether this is due to the fact that we haven't gotten the right dosage, we haven't put it on for the right duration, or that these types of neurons just simply aren't capable of having a permanent switch on of the paternal copy of UB3A. So it's an unsatisfying answer, but we don't know yet, and it's something that we're very actively pursuing because it's going to be really important as to both whether this could be brought forward as a therapeutic, and if so, how it would be treated and what the regulation and dosing schedule would be.
the next question was, what results, if any, or progress has come of the human trials in Tampa? This is the This is referring to the minocycline trial that I mentioned during my talk. I think we are, as I mentioned, we are, the preclinical studies from the Weber lab have not yet been formally reported or published. He has presented them in conference proceedings in a very preliminary fashion, but I don't have data from those from the preclinical data to review and analyze. And the clinical work, that trial has been completed, but the results are still, from my brief interactions with Ed prior to this meeting, they are not ready for release. He is hoping or anticipating releasing them hopefully in the fall, in the not too distant future. Any other comments from the panel on this?
I want to add one cautious note. I think, well, of course, we are all looking very much forward to see the results of the minocycline trial, but having run several large randomized controlled trials in our institute, I want to emphasize that for any trial that we did, for any disorder that we did, we see a huge placebo effect, and that is, or it's not even the placebo effect, that it means that the children that get the placebo will show a very significant improvement, even on objective outcome measures, even like on IQ tests. And the reason for that is, there's many reasons. For instance, they do the test twice, they feel more comfortable, the parents really want to see an improvement, and so they start to see an improvement. But of course also because we're looking at children and there's normal neurodevelopment, and every child, even an Angelman child, will improve over time even if you give it just 2 jelly beans a day. And I want to point out that for every study that you're going to do, which is going to be a single-arm trial and without a control, there will be an improvement. So, of course, we have to wait for the results, but I can hardly see any other result than this is going to give some positive results, and we really have to wait how that then is going to pan out in a large trial, in a controlled trial.
The next one was directed at Ben, and actually, Ben, this was a variation on this question. I think was presented was addressed by Ben during his talk. So, Ben, any further comments on the Rett Syndrome Foundation and your talk? This is where YouTube, his talk to the Rett Syndrome Foundation was posted on YouTube, as this talk will be. So in the modern social media world, we need to comment on this kind of thing. So Ben, please go ahead.
Yeah, just to reiterate, the reason we're looking at, and you might want to elaborate on this since it's a collaboration with you, the reason we're looking at critical windows of treatment is, one of the many reasons is to identify clinical endpoints that can be used in both preclinical and clinical trials, and also to know when the optimal age of intervention would be for treatment in Angelman syndrome. Also to know whether it would need to be lifelong or if it could be only during development. These are all really important questions, and in my mind, they're going to guide almost any preclinical or clinical trial that go forth from that.
Recently, there's been a report just, I guess it was last week. I saw it before I left town to come here about the reversal, or about in Down syndrome, which is trisomy 21, where there are three chromosome 21s in individuals with Down syndrome. A study where they had silenced, completely silenced one of those three chromosomes. Are any of you familiar with this study? It attracted quite a bit of press. Care to comment on its relationship to Angelman syndrome?
I mean, I think the goal of that study was to silence an entire chromosome, so I don't think an analogous approach would be useful for Angelman syndrome. What we really ideally want to do is to have something that's very specific for activating UB3A and not affecting other genes. So that's the goal of our research. So I don't think the approach can be quite analogous.
If detected during pregnancy, is there a way to alter the defective chromosome in utero?
Once again, this is where I think the preclinical studies come into play. So we have a compound that can unsilence the maternal copy of UB3A. It's something that in theory we can deliver to pregnant mice and see if it has an effect in utero on the pup that is then subsequently born. So this is something that we don't have an answer to, but it's something that we can address through preclinical studies.
Is there anything available in research for behavioral issues? My angel is 9 years old and he kicks, hits, and pulls others' hair, and it's getting worse. What causes this behavior and how can it be reduced? Any ideas? This is also preventing him from focusing, and he was doing so well. So, Jane, can you?
I would suggest that parents who are interested in this type of issue come to my talk tomorrow morning at 8:30. I'm going to be talking about the behavioral modules that were developed for parents to go to if they have questions about challenging behavior in their sons and daughters. And it's under the umbrella of applied behavior analysis approaches. So what we've tried to do is look at some of the reasons why challenging behavior develop. They're contributing factors as well. There's a reason why children act the way they do. That's called the functions of the behavior. So you have to understand what the child's trying to communicate through the problem behavior, find a more acceptable alternative way to teach them to communicate, and reward the good behavior as opposed to rewarding the behavior you want to get rid of. So there's a methodology, there's a framework for understanding and treating problem behavior as well as teaching skills to children with Angelman syndrome. And that's what we're going to be talking about tomorrow morning. So that might address some of the questions that were raised in this scenario.
So this is an area where, where there are best practices available that will be discussed extensively at this meeting. Last question on this slide. What is best when levopromazine does not work anymore, neither risperidone? This is, yes, I thought this might be the case. What's that?
I think Ron Hebert or Mark Mispacker should be.
Yeah, this is on sort of the, these are more for behavior issues more than epilepsy, yeah. Yeah, so I unfortunately, I don't think we have anybody on the panel with the clinical expertise to comment on this one, but there are people that are on site that I'll try to follow up with them and we'll post something online to address that question. Are any researchers focusing on the inhibitory-excitatory imbalance in Angelman syndrome? Several suffer from seizures as well as myoclonus jerks, Purkinje cells questions. Separately from this, do the library of compounds tried only look for turning on parental cells? Has anyone tried compounds in mice as therapeutic to relieve these symptoms in Angelman syndrome? So several kind of disconnected issues there, but I'll turn it over to the panel to.
So first of all, the drug screen we're looking at currently is only to find compounds that can turn on the paternal copy of UB3A. So we're not screening for anything else at that moment, because we think if we can get that approach to work, that that has the potential to provide the most benefit. We are very interested in the kind of imbalance in brain activity that occurs from the loss of UB3A, this so-called excitatory inhibitory imbalance, in which you can get a hyperactivity in the brain that can make one susceptible to seizures. And this is something we're studying very rigorously at a basic science level, and through that we hope that we might get some insight into therapeutics, but we're not currently screening therapeutics for that.
The next one is, will there be any further research on the calcium calmodulin kinase 2 discovery? As I mentioned in my talk, the Angelman Syndrome Foundation has funded, recently announced funding for a grant specifically to look at that. And Dr. Algorzma, was one of the people that was involved in that discovery. So maybe if you could comment on your
Yes,
on that
this
area.
question is still very much alive in our lab. I presented some of the work yesterday that we did and further reinforced the importance of CAM kinase in Angelman pathology. And definitely our lab will keep on working and see what the relation is and how we can go on that angle.
And I should add, there is a lot of research. I was in my talk talking about the work that the Angelman Syndrome Foundation has funded. There's a lot of other work going on that's being funded by the NIH and organizations in Europe and throughout the world on many aspects of these disorders. So there is a lot of other activity going on. Could Alzheimer's new medication help treat Angelman syndrome? It is a new compound named J147 and does not affect AMPA or NMDA receptors. Off the top of my head, I don't know J147. I'm guessing it's Johnson & Johnson possibly, but that's pure speculation from the J, but that's...
I have no insight.
No insight on that one.
No.
And one we will have insight on, has anyone done autopsy on Angelman syndrome mice brains? There are probably quite a few people in the room here that have done extensive autopsy work on Angelman syndrome mice brains.
A very typical thing that we do is study in great detail the mouse brains, both at different levels by using light microscopy, electron microscopy. Portia Kuntz in my lab is doing live 2-photon imaging. So we're imaging the brain in very many different ways and assessing it at very many different levels histologically. So yes, this is done extensively.
The next one, which are conditions to run as guinea pig and to which risks it exposes the patient? I think they're indicating sort of what are they, when a clinical trial is done, what are the risks that the patients are experiencing? Is my interpretation of that.
Well, this really depends on the drug, obviously, and the risk that we're willing to take really depends on the condition. So for instance, now we have a drug in clinical trial that we're giving to children that have intractable epilepsy and might even die from that epilepsy. So then you are willing to take much bigger risks than if it's just a drug to change behavior or aggression. So it really depends on the drug, and it really depends on the behavior or on the condition that you want to treat. So there's not a general rule.
So somewhat related to this is actually the one below that. What are the immediate risks distributing the active substance to humans with Angelman syndrome at this point? Now, my interpretation of active substance here is some of the compounds that are being that are in clinical studies are being considered for clinical studies. I think, I talked about the drug repurposing in connection. Those compounds Now, topotecan, because it's a cancer treatment, it is has some toxicity issues. That is not a drug that, from my understanding of it right now, I would be comfortable treating my child with, or anyone with long term, and it generally wouldn't be used that way. But minocycline, generally we don't treat people with antibiotics long term, and there would be some expected disruption of issues because of the basically bacteria in their body would no longer be active, and wipe those out, as well as possibly affecting MMP9 and doing things. But most of the other drugs, because they are FDA approved, we know their toxicity. We know they've already gone through phase 1 trials, which is generally where you're looking for toxicity effects and other things. So their safety profile is known. So the risk of the trials, generally for the ones that we've moved forward with, from that perspective is low. The risk is primarily because they haven't been used extensively in a long-term trial with kids with Angelman syndrome who have a different profile. They have a different genetic composition than neurotypical individuals. The other risk is that we simply will get a negative result or no result, right? That it will not have activity. So we don't just want to, we want them to be tested in a controlled environment so that we can monitor and assess whether they do work and whether they're effective before we just tell the public, hey, this is going to be useful and beneficial for your child. We want to know that they work and that they are safe and effective, that they're both safe and effective. Okay, care to expand on that?
I would just say that with compounds there might be some expected possible side effects that will be very closely monitored, but regardless of the clinical trial, any adverse event would have to be reported in a clinical trial.
Adults with Angelman syndrome often exhibit disruptive behaviors that make it impossible for a normal social life. Is it being investigated and getting a new drug to help control this type of behavior that are not antipsychotics because of the unwanted side effects? Maybe Jane, could you?
I'm not sure about the medication end of things. I know that using behavioral techniques and working with families to discover the reasons why these problem behaviors are occurring and trying to set up the environment to promote the development of more appropriate skills and trying to decrease the impact of the negative ones is the approach I would take. Now, I often work with psychiatrists too. If they do prescribe medication, a behavior analyst, for instance, can help with the tracking of the response to that medication through looking at very specific behavioral changes. So sometimes it's not an either-or question. It's how do we work together to see if we're getting a good effect in these types of situations.
I think the next one we'll skip. Human trials are underway in various aspects that we discussed. Are there any other models than mice in the making? I think this is a question of what other model organisms are there are there any other model organisms to work with?
So there are the flies. I know that several labs work on the flies.
Yes.
But I think most importantly right now, as we've heard also today, there are the human iPS cells. So we can make from skin cells, we can make neurons, and in several laboratories, including ours and Ben's, these are not going to be used as a model. So human is the next model, human cells. Yeah.
The next question is, any progress have come from the levodopa trial? Wen-Han, you're very involved in this study.
So we are still recruiting participants for the levodopa study because it is a double-blind, placebo-controlled study. We cannot unblind the study until we have enrolled everyone. We will not have any results probably for another year.
The next one is a question that came up from one of the sessions earlier today. What are the pros and cons of Angelman syndrome being associated with autism? Anyone want to comment on that?
Well, I think there's a number of pros. Part of what I look at is, are there studies or is there information in relation to autism that could be applied in helping people with Angelman syndrome? So there's techniques, there's different things we might do that are effective in people with autism that could be applied to the Angelman group in terms of dealing with problem behaviors and teaching more appropriate social behaviors. I understand in some places, insurance is tied into a diagnosis of autism, or help through the school system is linked to a diagnosis of autism, and parents are getting turned down for help because their son or daughter doesn't have a diagnosis. To me, that's a con, and for insurance agencies and school boards to think that you have to have a diagnosis of autism to benefit from behavioral and analytic techniques, I think that's a fallacy, and I think that does a disservice to other people who could benefit. So that's maybe a con to not having a diagnosis of autism and funding bodies requiring that diagnosis. But I think there's lots of pros too in terms of understanding more about some of the features of Angelman that overlap with autism, and maybe there's treatments that could be applied in the population that would be helpful.
I'm out of questions. Any questions from the audience? Going once, going twice, sold. I will be here all week. If anyone has any questions, please find me or link with me, connect with me. I'm happy to try to answer any questions or try to refer you to people that can. I want to thank the panel for their contributions. I want to especially thank Ben for his presentation, and thank you for staying so late. Let's go party.